Biochemistry is the quiet pulse beneath all living things. It is the study of the subtle, invisible chemistry that sustains every breath, every movement, and every moment of awareness. Beneath the skin, beyond the visible world of organs and tissues, exists a realm of constant motion, molecules joining and parting, atoms rearranging, and reactions taking place with precise rhythm. These tiny events form the foundation of life, though they pass in perfect silence. To study biochemistry is to listen closely to that silence, and to realize that within it lies the entire story of existence. At its heart, biochemistry unites two great sciences: biology, the study of life, and chemistry, the study of matter and its transformations. Together they describe not only what living things are made of, but also how they function at the most fundamental level. Every heartbeat, every neuron signal, every blade of grass turning toward the sun is a biochemical event. Molecules interact in finely tuned sequences, energy flows through them, and structures assemble or dissolve according to rules that have operated since the dawn of life on Earth. And yet, though governed by the same physical laws as inanimate matter, these molecular interactions create something extraordinary, a living system capable of reproducing, adapting, and thinking. Imagine drifting through the calm interior of a single living cell. The boundaries of the cell are delicate but precise, a thin membrane formed of lipids and proteins that separates the inside from the world beyond. Within it, the space is not empty, but filled with a gentle, shimmering crowd of molecules. Water molecules slip smoothly past one another, carrying dissolved ions and nutrients. Long chains of carbon-based compounds twist and fold into intricate shapes. Tiny packets of enzymes float purposefully through the cytoplasm, catalyzing reactions that build or break down other molecules. This inner landscape, invisible to the eye, is a world of order amid seeming chaos, of quiet equilibrium, sustained by endless motion. What makes life different from non-life is not the materials it contains, but the way those materials are organized. The atoms found in a leaf or a human being are the same atoms that exist in the air and the sea. What changes is the pattern, the choreography of how those atoms are arranged and rearranged. In the chemistry of life, patterns matter. Biochemistry concerns itself with these patterns, how they form, how they are maintained, and how they can be gently altered by energy and environment. It asks how molecules come together to form membranes, how they build enzymes capable of transforming energy, and how they store instructions in the delicate spirals of DNA. Life depends on the delicate interplay between stability and change. Each cell must be stable enough to persist, to preserve its identity across time, yet it must also be fluid and responsive, able to adapt to subtle changes in temperature, light, or nutrient supply. Biochemical reactions provide that adaptability. They proceed in networks pathways that interconnect and regulate one another. If the energy in one part of the system falters, another pathway compensates. If a harmful molecule appears, enzymes neutralize it. The living cell survives because its chemistry is both disciplined and flexible, a dynamic equilibrium that continues hour after hour without pause. At this microscopic level, even simple events have beauty. When a molecule of glucose enters a cell, it does not simply burn for energy. Instead, it passes through a serene sequence of steps, each one guided by a specific enzyme. Energy is released not in a burst, but in small, carefully measured amounts, stored in high-energy compounds such as ATP. This energy, in turn, powers other reactions, the construction of proteins, the repair of membranes, or the replication of DNA. The entire cycle unfolds like a well-rehearsed piece of music, with each enzyme contributing a note, each molecule passing energy to the next in an unbroken chain. Although biochemistry is deeply technical when studied in the laboratory, it also has a quiet, poetic side. It reveals how deeply connected all living things are. The enzymes in a human liver are not fundamentally different from those in a blade of grass or a mushroom. The same molecules of ATP that provide energy to a human heart also drive the motion of bacterial flagella and the opening of plant stomata. Life on Earth, from the smallest microbe to the largest whale, shares a single biochemical language. That unity speaks of a common origin, a time billions of years ago, when simple molecules first learned to copy themselves and to harness energy from their surroundings. To the biochemist, life is not a miracle of chance, but the inevitable outcome of chemistry, given enough time and the right conditions. When the young Earth cooled, when water gathered in shallow pools and carbon compounds drifted through the air, something subtle began to happen. Simple molecules collided and joined, forming chains that could store energy or react with others. These interactions were random at first, but as certain reactions proved more stable, they persisted. Over vast stretches of time, these reactions became organized, enclosed within membranes, and capable of reproducing themselves. The first cell was born, not as a sudden leap, but as a gradual weaving together of chemical threads, a process that continues unbroken to this day within every living being. In modern times, biochemistry allows us to understand life with both precision and awe. It explains how oxygen is transported by the red pigment hemoglobin, how the brain sends signals through neurotransmitters, and how light striking a leaf sets off the chain reaction we call photosynthesis. Yet behind every explanation lies the same simple truth. Life is chemistry that has learned to sustain itself. Our bodies are not alive on their own, but together, through balance and feedback, they create something that is. To comprehend biochemistry, therefore, is not merely to learn reactions and formulas, but to glimpse the deep order of nature itself. When one considers the scale of these reactions, there is a sense of calm humility. Within a single human cell, thousands of biochemical processes occur every second, each dependent on countless others, each maintaining the cell's internal balance, and yet we feel none of it. We breathe, move, and think without awareness of the molecular symphony unfolding inside us. The chemistry of life proceeds quietly, like a hidden rhythm beneath consciousness. It is steady, precise, and endlessly patient, maintaining its dance from the moment of birth until the final breath. Perhaps this is why biochemistry lends itself so naturally to contemplation. It teaches that complexity can arise from simplicity, that harmony can exist without supervision, and that beauty may be found even in the unseen. It reminds us that our bodies, our thoughts, and our emotions are all sustained by the same elegant chemical principles that guide a coral reef or a blade of grass. We are not separate from the chemistry of nature. We are its expression. As we move deeper into this lecture, we will explore the building blocks that make this quiet science possible: the atoms, the molecules, and the macromolecules that sustain every living form. But before we journey into those details, it is enough to pause and acknowledge the gentle wonder of it all. Every heartbeat, every whisper of air in the lungs, every spark of thought in the brain arises from a choreography of molecules obeying simple laws with exquisite precision. To study biochemistry is to stand in quiet reverence before that truth, to see life not as a mystery to solve, but as a balance to understand. Within each cell, in every corner of living matter, chemistry hums softly, tirelessly, and beautifully, reminding us that even the smallest things can hold the essence of life itself. 2. The origins of biochemical thought. Before biochemistry became a recognized science, there existed only wonder. For countless generations, humans gazed at living things, plants sprouting from soil, animals breathing and moving, the human body pulsing with vitality, and asked what animated them. Long before microscopes or laboratories, there was only the quiet observation of life and the intuition that something unseen guided it. Ancient philosophers called this animating principle the vital force, a mysterious energy thought to separate the living from the non-living. To them, life could not possibly be reduced to material components or mechanical processes. It had to be something more, something sacred, immaterial, and beyond the grasp of ordinary matter. In the early civilizations of Egypt, India, Greece, and China, ideas about life were deeply intertwined with philosophy and spirituality. The body was seen as a vessel of harmony, governed by balances of elements or energies: air, water, fire, and earth in Greek thought, or the forces of yin and yang in Chinese tradition. These early systems were not scientific in the modern sense, but they represented humanity's first attempts to describe the chemistry of life in metaphorical terms. When physicians of ancient Greece spoke of balancing humors or maintaining bodily equilibrium, they were intuitively describing what modern science would later recognize as homeostasis, the self-regulating stability of living systems. It was not until the late Middle Ages that natural philosophy began to shift toward experimentation. Alchemists, often dismissed as seekers of gold, were in truth early chemists who observed how substances changed when heated, mixed, or dissolved. Their work laid the groundwork for modern chemistry, even if their aims were mystical. Through their trials, they learned that matter could be transformed, and that these transformations followed consistent patterns. The alchemists' furnaces and flasks were the ancestors of today's laboratories, and in their pursuit of the elixir of life, or the philosopher's stone, they unwittingly began to probe the boundaries between the living and the inert. By the 17th century, as science entered its age of reason, the boundaries between chemistry and biology began to blur. Pioneers such as Robert Boyle and Antoine Lavoisier brought rigor to the study of matter, describing elements and reactions in measurable terms. Lavoisier, often called the father of modern chemistry, recognized that combustion and respiration shared a common principle. Both involved the combination of substances with oxygen. In his careful experiments, he measured how animals consumed oxygen and produced carbon dioxide, concluding that respiration was, in essence, a slow form of burning. This was one of the earliest moments when chemistry was directly linked to the processes of life. Still, many thinkers of the 18th century clung to the idea of vitalism, the belief that living matter possessed a special essence, absent in non-living things. They argued that the processes of life could not be explained by ordinary chemistry. For a time, this belief seemed reasonable. When one observed a seed sprouting into a tree or an animal growing from a single fertilized egg, it was difficult to imagine that such complexity could arise from simple molecules. Life seemed to contain a purpose, a direction, something that mere chemical laws could not capture. And so, vitalism persisted, comforting in its mystery. The turning point came in the early 19th century, when a quiet revolution took place in a small German laboratory. In 1828, a young chemist named Friedrich Wöhler succeeded in synthesizing urea, a compound previously thought to exist only in living organisms, from entirely inorganic materials. This discovery was simple yet profound. It showed that a substance produced by the body could also be made by purely chemical means, without any vital force at all. The implications rippled through the scientific world. If one organic compound could be created in the lab, perhaps all of life's chemistry could eventually be understood in terms of ordinary physical laws. Vitalism began to lose its hold. In the years that followed, chemistry turned its attention increasingly toward biological substances. Scientists began isolating and identifying compounds from plants and animals, sugars, fats, proteins, and enzymes, and learning how they functioned. The microscope, refined through the 19th century, opened new worlds within the cell. Robert Brown observed the nucleus. Matthias Schleiden and Theodor Schwann proposed that all living things are composed of cells, and Rudolf Virchow later added that every cell arises from another cell. Biology was no longer merely descriptive. It was becoming mechanistic, rooted in chemistry and physics. By the mid-19th century, Louis Pasteur brought further clarity to the chemical nature of life. Through careful experiments, he showed that fermentation, the transformation of sugar into alcohol, was not a simple chemical reaction, but was carried out by living microorganisms, specifically yeast. He also demonstrated that life did not spontaneously arise from non-living matter, as many had once believed. Pasteur's work deepened the connection between biology and chemistry, showing that tiny living agents could drive enormous transformations, both in the laboratory and in nature. The invisible world of microbes, once unknown, became the foundation of both biochemistry and medicine. Around the same time, a German chemist named Eduard Buchner performed an experiment that would bridge the gap between life and chemistry even more completely. In 1897, he crushed yeast cells and filtered out their contents, creating an extract devoid of any living organisms. To his astonishment, the cell-free liquid still converted sugar into alcohol. This meant that the process of fermentation was driven by chemical agents, enzymes that could function outside the living cell. With this discovery, the age of modern biochemistry had truly begun. Life's processes could be studied not only within living beings, but also in test tubes through their chemical components. As the 20th century unfolded, the science of biochemistry blossomed. Researchers began to map out metabolic pathways, tracing how energy flowed through cells in cycles and chains of reactions. The discovery of vitamins revealed the subtle molecules that enable these reactions to proceed efficiently. Scientists such as Hans Krebs identified the citric acid cycle, showing how nutrients are broken down to release energy. Others discovered how light energy drives photosynthesis, linking the chemistry of plants and animals in a single grand cycle. Each finding reinforced the idea that life was a chemical network of extraordinary complexity, yet governed by elegant simplicity. The next great leap came with the discovery of the structure of DNA in 1953 by James Watson and Francis Crick. With the help of Rosalind Franklin's X-ray images, they revealed the double helix, two strands intertwined, carrying the genetic code of life. This discovery bridged biochemistry and genetics, revealing that the instructions for building and maintaining living organisms are themselves chemical in nature. The study of molecules had come full circle, from ancient wonder about the vital force to the realization that life's essence resides in the precise arrangement of atoms. Today, biochemistry stands at the heart of modern science. It underpins medicine, genetics, pharmacology, and even the study of consciousness. Yet its origins remain rooted in a simple curiosity. What is life made of? The journey from mysticism to molecular clarity took centuries, each generation adding a small piece to the puzzle. And through it all, one truth has endured: that the beauty of life lies not in defying nature's laws, but in expressing them with breathtaking complexity. When we reflect on this long history, there is something humbling about how it mirrors life itself. Just as biochemical reactions evolve from simple to intricate forms, so too did our understanding of them. Each discovery built upon the previous one, forming a chain of knowledge as seamless as the reactions within a cell. The earliest philosophers sensed that life was special. They were not wrong, only imprecise. The mystery they perceived still exists, but it now rests not in a supernatural force, but in the astonishing order of molecules, the quiet perfection of nature's chemistry. In essence, the origins of biochemical thought remind us that knowledge grows in layers, that understanding deepens slowly, and that life's mysteries reveal themselves only to those who observe patiently. The same patience that guides a scientist's hand at the laboratory bench is reflected in the patience of nature herself, in the slow assembly of molecules, in the steady unfolding of evolution, and in the calm persistence of life across billions of years. Biochemistry began as a whisper of curiosity, and became a language of truth. It tells us that the living world is not separate from the material one, but arises naturally from it, shaped by laws both simple and profound. And as we look back on the centuries of discovery that brought us here, we begin to see that every question about life, no matter how ancient, was really a question about chemistry waiting to be answered in its own time. 3. The elements of life. All living things, no matter how vast or small, are built from a remarkably simple foundation of elements. Whether it is the soft tissue of a leaf, the delicate structure of a coral reef, or the intricate anatomy of a human being, every form of life draws from the same universal palette of matter. Out of the 100 or so naturally occurring elements found on Earth, only a handful form the essence of all biology. These are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, often abbreviated as CHNOPS. Together, they account for nearly all the atoms within every living cell, weaving the chemistry of existence from an elegant and limited vocabulary. The simplicity of these ingredients is part of their power, for from their combinations arise all the complexity and beauty of life. If one were to look deeply into the structure of any organism, it would become clear that these elements do not merely coexist, but cooperate. They join in bonds, form molecules, and interact through forces that balance strength with flexibility. Their behavior follows precise physical laws, yet those laws allow for endless variety. The molecules they form can be simple, like water or carbon dioxide, or astonishingly complex, like DNA or a folded protein with thousands of atoms arranged in perfect geometry. In this way, biochemistry reflects one of nature's favorite patterns: from the few, the many. The first and most essential of these elements is carbon. It is the backbone of all organic molecules, the quiet architect of living matter. Carbon's power lies in its versatility. Unlike most elements, it can form 4 bonds at once, linking to itself or to other atoms in chains, rings, or branches of almost limitless variation. This ability gives carbon a flexibility unmatched by any other element. It can build simple gases such as methane, the smallest organic molecule, or immense macromolecules that store genetic information and energy. Because of its bonding properties, carbon provides stability and strength, yet remains adaptable, the perfect material for life's ever-changing chemistry. Without carbon, no cell could exist, no tissue could form, and no organism could evolve beyond a mere chemical curiosity. Next comes hydrogen, the simplest and most abundant element in the universe. It was among the first atoms to form after the birth of the cosmos, and it continues to play a vital role in sustaining life. Within living systems, hydrogen rarely stands alone. It bonds with oxygen to create water, the universal solvent that fills every cell and carries nutrients across membranes. It binds with carbon to form hydrocarbons, which store and release energy in metabolic reactions. Hydrogen's behavior defines acidity and alkalinity through its protons, determining the delicate pH balance upon which countless biochemical reactions depend. Its presence is subtle but constant, an invisible thread connecting the chemistry of the stars to the chemistry of the cell. Oxygen, too, plays a profound role. It is both a giver and a taker of energy, essential to the quiet combustion that fuels life. Within our cells, oxygen acts as the final electron acceptor in the chain of respiration, allowing energy to be extracted efficiently from food molecules. Without oxygen, that process would falter, and complex life would suffocate in an instant. Yet oxygen is also a builder. It forms part of water and countless organic compounds, linking with hydrogen, carbon, and nitrogen to form alcohols, acids, and sugars. Its high reactivity makes it both vital and dangerous, capable of sustaining life or damaging it through oxidation. In the balance between oxygen's generosity and volatility, evolution has crafted delicate protective systems, antioxidant molecules that guard against the harm caused by oxygen's reactive forms. Nitrogen, the next essential element, is the cornerstone of growth and renewal. It forms the backbone of amino acids, the building blocks of proteins, and of nucleotides, the components of DNA and RNA. Though nitrogen gas fills most of Earth's atmosphere, its molecules are too stable to be used directly by most organisms. Instead, certain bacteria and archaea have evolved to fix nitrogen, converting it into ammonia and related compounds that plants can absorb. From there, nitrogen moves through the food chain, passing from plant to herbivore to predator, eventually returning to the soil through decay. In this quiet cycle, nitrogen binds the living world together. It is the element of connection, linking the invisible air above to the visible abundance of life below. Phosphorus, though less abundant, is no less vital. It forms the structural backbone of nucleic acids, joining sugar molecules to create the long chains of DNA and RNA. It is the key component of ATP, the molecule that carries energy within every cell. Each time a cell moves, divides, or repairs itself, it draws upon the energy stored in the bonds of phosphorus-containing compounds. Without this element, energy could not be harnessed efficiently, and the delicate machinery of metabolism would come to a halt. Phosphorus also contributes to the strength of bones and teeth, binding with calcium to form the sturdy mineral structures that support complex organisms. It is, in a sense, both the power source and the framework of life. Sulfur completes this small but mighty group. It is found in several amino acids and in certain coenzymes that facilitate metabolic reactions. Though present in small amounts, sulfur adds stability and reactivity where needed. It helps proteins maintain their shape through disulfide bridges, those subtle bonds that hold folded chains in their correct configuration. The faint scent of sulfur often marks the presence of biological activity, from volcanic vents on the ocean floor to the decomposition of once living tissue. In the language of life, sulfur's signature is quiet but distinct, a reminder that even the most delicate reactions may depend on the smallest contributions. Beyond these 6 primary elements lies a supporting cast of trace elements, minerals such as iron, calcium, magnesium, zinc, and potassium, each playing its part in the subtle choreography of living systems. Iron carries oxygen within the blood, cycling between its oxidized and reduced forms in a rhythmic exchange. Calcium strengthens bones but also serves as a messenger, triggering muscle contractions and nerve impulses. Magnesium stabilizes the energy molecule ATP, ensuring that its power can be used precisely where needed. Potassium and sodium regulate electrical signals across membranes, allowing cells to communicate. Each of these elements, though present in minute amounts, adds to the stability and responsiveness that define life. Their balance is delicate. Too little or too much of any one can disturb the body's harmony, reminding us that nature values moderation above all. When we think of life in this elemental way, it becomes clear that the boundary between the living and non-living is less absolute than it seems. The same carbon found in a falling leaf also exists in ancient rocks and drifting stardust. The oxygen we breathe today once belonged to water in primordial oceans. The nitrogen in our muscles may have passed through the roots of plants or the atmosphere of another continent centuries ago. Life is not built from new matter, but from matter endlessly recycled, reshaped into new forms again and again. The atoms within our bodies are older than the earth itself, forged in the cores of ancient stars and scattered through space before finding their way into living systems. In this sense, biochemistry is not merely a science of life, but also a bridge between the cosmos and the cell. Each element, from the humblest hydrogen to the intricate lattice of phosphorus and sulfur, tells part of this greater story. Their bonds are not random. They follow rules, written into the very nature of matter. Yet the result of those rules is a world capable of awareness and imagination. To reflect on the elements of life is to recognize that our thoughts, our emotions, and even our sense of wonder are built upon the simplest materials of the universe. Out of atoms that once drifted aimlessly through space, a consciousness has arisen that can look back upon the stars and ask how it came to be. In the end, the elements of life remind us of unity, that all living things share not only a planet but a chemical kinship, from the algae floating in a pond to the human heart beating quietly through the night. The same elements flow through every vein, every root, every breath. They combine, separate, and combine again in an endless cycle of renewal, their forms changing, but their essence eternal. And in that constancy lies something profoundly calming, the assurance that even as life evolves, ages, and fades, the elements endure patient, quiet, and everlasting, waiting to be shaped once more into the miracle of living form. 4. The role of water. Water is the quiet medium in which all life unfolds. It is so common, so familiar, that we often overlook its extraordinary nature. Yet without it, life as we know it could not exist. Every living cell is, at its core, a small ocean. Within that ocean, molecules drift, reactions take place, and energy flows. Water surrounds every protein, cushions every membrane, and carries every signal. It is not just a background or a solvent. It is an active participant in life's chemistry, shaping and sustaining the reactions that define existence. To understand biochemistry is to understand the profound and delicate role of water. A single water molecule is simple, made from 2 atoms of hydrogen and 1 of oxygen. Yet in this simplicity lies perfection. The oxygen atom draws electrons closer to itself, giving it a slight negative charge, while the hydrogen atoms become slightly positive. This imbalance makes water a polar molecule, with a positive side and a negative side, capable of attracting other charged or polar molecules. When water molecules meet, they form hydrogen bonds, gentle, fleeting attractions that constantly form and break. Each molecule can bond to several others, creating an intricate network that shifts like a living fabric. Though each bond is weak, together they create a structure both stable and fluid, an elegant balance that underlies all of life's chemistry. Because of these interactions, water behaves unlike almost any other substance. It remains liquid over a wide range of temperatures, resists sudden changes in heat, and expands when it freezes, an anomaly that allows ice to float rather than sink. These properties are not mere curiosities. They are essential for life. The oceans do not freeze solid because ice insulates the liquid beneath it. The bodies of animals do not boil under the summer sun because water absorbs heat slowly and releases it gradually. This remarkable capacity to moderate temperature grants the planet its gentle climate and gives living organisms a stable internal environment in which biochemical reactions can proceed without chaos. Within cells, water performs its miracles quietly. It dissolves a vast variety of substances, earning its title as the universal solvent. Salts, sugars, amino acids, and nucleotides all separate into their charged components when surrounded by water molecules. These dissolving processes allow the molecules of life to move freely, meet one another, and react. In a way, water is the stage on which all of life's chemical dramas unfold. The folding of a protein, for example, depends on how its parts interact with water. Some portions of the molecule are drawn to it, others avoid it, turning inward to escape the liquid's reach. In this way, water dictates the 3-dimensional shapes that determine the function of every enzyme and structural molecule in the body. Even beyond the molecular level, water exerts its influence. It forms the basis of bodily fluids, from the plasma in blood to the cytoplasm in cells. It transports nutrients and waste, carries hormones to their targets, and lubricates joints and tissues. In plants, water flows upward through narrow vessels in defiance of gravity. driven by cohesion and adhesion, the gentle attraction between water molecules and between water and the surfaces they touch. This steady flow nourishes every leaf and blossom, connecting the soil to the sky in an unbroken column of liquid life. When the sun warms a leaf, water evaporates, pulling more upward behind it, a silent and continuous heartbeat for the plant. The importance of water extends far beyond biology into the very origin of life itself. Long before organisms took shape, water was already acting as a cradle for chemistry. The early Earth was a world of oceans and vapor, a planet wrapped in storms and lightning. In those waters, simple molecules began to interact, forming amino acids, sugars, and nucleotides, the precursors to living matter. Water provided the medium in which these reactions could occur, protecting fragile compounds from destruction while allowing them to collide and combine. The tides, the waves, and the gentle cycles of evaporation and condensation may have been the rhythms that gave birth to the first cells. Even today, every embryo develops in a watery environment, echoing those ancient beginnings. Water's role in living systems is both chemical and physical. It participates directly in many reactions, not merely as a backdrop, but as an ingredient. In hydrolysis, for example, water breaks larger molecules into smaller ones, a process vital for digestion and metabolism. In condensation reactions, water is produced as smaller molecules join together to form larger ones, such as when amino acids link to create proteins or when sugars bond to form starch. Through these alternating acts of giving and taking, water mediates the balance of construction and destruction that keeps life in motion. It is both the sculptor and the solvent, shaping and reshaping the materials of living form. On the microscopic scale, the behavior of water molecules governs how substances move within the cell. Diffusion, the passive spreading of molecules from high concentration to low, depends entirely on water's fluid nature. So does osmosis, the movement of water itself across membranes to equalize concentration. These simple physical processes maintain the balance of salts and nutrients that every cell requires. When water flows into a plant cell, it creates pressure against the cell wall, giving leaves their firmness and stems their strength. When it leaves, the plant wilts. In animals, similar shifts of water regulate blood pressure, nerve function, and even thought. Every moment, water molecules drift through membranes, slipping through narrow channels, responding to subtle differences in concentration and charge. They act without sound or pause, sustaining the constancy that we call life. Perhaps what makes water most remarkable is its ubiquity. It makes up most of the human body by weight, and an even greater proportion of the simplest organisms. It covers nearly 3/4 of the planet's surface and cycles endlessly through the atmosphere, the soil, and the seas. The water we drink today is ancient. It has rained, frozen, flowed, and evaporated countless times across millions of years. It has passed through rivers, clouds, and living beings, returning again and again to nourish life. Each molecule may have once been part of an ocean, a glacier, or even another organism. In every drop, the memory of the Earth's long history is preserved, reminding us that we are part of a cycle far greater than ourselves. Water is also a quiet carrier of beauty. It gathers light, bending and scattering it into color. It amplifies sound, softens landscapes, and nourishes growth wherever it flows. Its presence brings calm, and its absence brings desolation. In the human body, it keeps the eyes clear, the skin supple, and the brain suspended in gentle cushioning. In rivers, it sculpts valleys. In clouds, it shapes the sky. The same molecule that glistens on a leaf at dawn also flows in the bloodstream of every creature, joining all forms of life in a shared chemistry of renewal. The deeper one contemplates water, the clearer its mystery becomes. It is a compound so simple in composition, yet so complex in behavior. Its ability to dissolve, to stabilize, and to moderate temperature gives it properties that seem almost designed for life. Yet these properties arise naturally from the laws of physics, from the angles of its bonds, the distribution of its charges, and the geometry of its molecules. In this sense, water is a reminder of the quiet intelligence of nature, that the same forces which shape stars and planets can also give rise to tenderness, growth, and sentience through a molecule so unassuming it covers our world unnoticed. To reflect on water is to recognize that it is both the stage and the actor, both the vessel and the force. Every heartbeat is carried by its pulse, every thought relies on its movement, every seed depends on its gentle touch. In all of its forms, solid, liquid, or vapor, it sustains the rhythm of life. When rain falls, it returns to the soil the same molecules that once flowed through ancient seas. When we drink, we become part of that unending circulation, taking into ourselves the history of the planet. Water teaches stillness through its motion and balance through its change. It reminds us that softness can sustain strength, that the gentlest substance can shape mountains and cradle entire worlds. Within each cell of every living being, water continues its ancient work, quiet and unseen, yet utterly essential. It is the silent partner of life, patient, enduring, and endlessly forgiving, carrying the memory of all things that have ever lived, and the promise of all things yet to come. 5. Carbon's infinite creativity. In the vast language of chemistry, carbon is the quiet poet, composing endless variations of form and function. It is the element that gives life its architecture, the one that allows complexity to blossom from simplicity. Though it sits modestly among the periodic table's middle ranks, carbon possesses a rare and extraordinary talent, the ability to bond with itself and with others in nearly limitless ways. From this single gift comes the astonishing diversity of living matter, the intricate spiral of DNA, the delicate shape of a leaf's pigment, the resilience of bone, and the invisible molecules that make thought and memory possible. To study carbon is to gaze into the very heart of life's design, where order and creativity meet in perfect balance. At the atomic level, carbon's versatility stems from its 4 valence electrons, 4 tiny possibilities waiting to be shared. This number allows it to form up to 4 covalent bonds, connecting not only with other elements but also with itself, creating long chains, branching networks, and closed rings. Unlike many other atoms that form rigid or limited structures, carbon's bonds are stable yet flexible, allowing for both strength and variety. In a sense, carbon behaves like a patient builder, able to construct everything from the simplest shelters to the most elaborate palaces of molecular architecture. Each bond is a deliberate step, each connection a piece of an elegant design that has been refined by evolution for billions of years. In its pure forms, carbon already demonstrates this creative range. As graphite, it lies soft and dark, its layers of atoms sliding smoothly across one another, capable of leaving marks on paper and conducting electricity. As diamond, it becomes hard and brilliant, each atom locked in a perfect 3-dimensional lattice of unbreakable strength. These extremes, pliant and strong, opaque and radiant, reveal the duality of carbon's nature. The same element that etches our words can also refract light into dazzling clarity. In biological systems, this same duality persists. Carbon forms molecules that are both delicate and resilient, transient and enduring. Its bonds are strong enough to support the intricate scaffolding of life, yet not so strong that they cannot be rearranged when needed. From carbon's endless combinations arise the essential molecules of biology: carbohydrates, lipids, proteins, and nucleic acids. Each of these great molecular families begins with the same foundation chains and rings of carbon atoms, but each takes on its own character through subtle variations in bonding and structure. In sugars, carbon forms open chains that can curl into rings, capturing energy from the sun and storing it in accessible chemical bonds. In lipids, it arranges itself into long hydrophobic tails that resist water, creating membranes that separate the inner world of the cell from its surroundings. In proteins, it becomes the backbone that links amino acids into sequences of endless possibility, each folding into a precise three-dimensional shape. And in nucleic acids, it forms the framework for genetic information, the delicate spirals of DNA and RNA that encode the instructions for life itself. The secret to carbon's infinite creativity lies in its ability to combine stability with adaptability. Its single bonds are flexible, able to rotate freely, allowing molecules to twist and turn into countless shapes. Its double and triple bonds, though stronger and more rigid, add contrast and function, introducing regions of tension and reactivity. These variations in bonding allow carbon molecules to respond sensitively to their environments. They can store energy and release it, capture light and convert it, or form surfaces that interact selectively with other molecules. Through these dynamic relationships, carbon makes possible the subtle responsiveness that defines living systems, the capacity to sense, adapt, and evolve. Carbon's creativity extends not only to structure, but also to symmetry and pattern. Many biological molecules are chiral, meaning they exist in mirror image forms, like left and right hands. This asymmetry arises from the tetrahedral geometry of carbon's bonds, where 4 different attachments create 2 non-superimposable configurations. Life has chosen one of these orientations, almost exclusively a mysterious preference that adds another layer of order to the natural world. Every amino acid in the proteins of living organisms shares the same handedness, and every sugar follows the opposite. This uniformity allows the molecular machinery of life to fit together with astonishing precision, as if all of its parts were carved by the same artisan. Yet for all its versatility, carbon remains modest. It does not seek dominance. It simply forms bonds wherever conditions allow. In the cold darkness of interstellar clouds, carbon atoms attach to hydrogen, oxygen, and nitrogen to form simple organic compounds, drifting through space as potential seeds of life. On early Earth, similar compounds accumulated in the oceans and atmosphere, combining into more complex molecules. Through the gentle persistence of chemistry, they formed the precursors of proteins and nucleic acids. Even before life existed, carbon was already practicing its art building, breaking, and rebuilding, exploring every possible arrangement until something new and self-sustaining emerged. What makes carbon unique among elements is its ability to serve as both the skeleton and the storyteller of life. Its chains form the physical structure of living matter, while its arrangements encode function and meaning. In DNA, sequences of carbon-based nucleotides record information, guiding the construction of enzymes and tissues. In hormones and neurotransmitters, small carbon molecules carry signals that influence growth, emotion. In pigments and photosynthetic compounds, carbon absorbs light and transforms it into energy. Each role is different, yet all are connected by the same simple element, repeating its bonds in infinite variation. It is as though carbon writes the script of existence using only a few letters, arranging them into a narrative that spans all of biology. The stability of carbon compounds also ensures life's continuity. Many of the molecules in our bodies have lifetimes that extend for days or weeks, maintaining their form until the cell is ready to renew them. At the same time, carbon's bonds are not so unyielding that they resist change. Enzymes, the catalysts of life, can rearrange them with gentle precision, allowing reactions to proceed smoothly at the mild temperatures of living organisms. Without carbon's balance between strength and flexibility, life would either freeze into rigidity or dissolve into chaos. Instead, it thrives in the middle ground, where order and motion coexist in harmony. Even beyond Earth, carbon's fingerprints mark the potential for life elsewhere. In the atmospheres of distant planets, astronomers detect methane and other organic compounds, signs that carbon's chemistry operates throughout the cosmos. Meteorites that have fallen to Earth contain amino acids and other carbon-based molecules formed in space, suggesting that the universe itself is predisposed to create the building blocks of biology. If life exists beyond our planet, it is likely built upon the same foundation, not by coincidence, but by the quiet inevitability of carbon's properties. To reflect on carbon is to appreciate how life arises not from chance, but from the inherent possibilities of matter. It reminds us that complexity need not be imposed from above. It can grow naturally from the interactions of simple elements under the guidance of physical law. The same principle that allows carbon atoms to form a diamond also allows them to form the fragile strands of DNA that encode consciousness. The same bonds that create the hardness of coal can also build the softness of a leaf or the translucence of an eye. There is a quiet poetry in that unity, a recognition that all living things share not only a common ancestry but a common chemistry. In the grand cycles of nature, carbon moves endlessly between earth, air, and life. It resides in the minerals beneath our feet, the carbon dioxide in the atmosphere, and the living tissues that breathe it in and out. Plants capture it through photosynthesis, converting sunlight into sugar. Animals consume those sugars, transforming them into energy and structure. When life ends, carbon returns to the soil and air, ready to begin again. It is never lost, only transformed, participating in the eternal rhythm of creation and renewal. Every breath we exhale carries molecules that may one day nourish a forest or return to our lungs in another time. Carbon's infinite creativity is thus more than a chemical fact. It is a reflection of the continuity that binds all existence. From the first spark of life in ancient oceans to the beating of a human heart, carbon's bonds have held the story together, unbroken and ever evolving. It is the artist of matter, the patient architect of complexity, and the silent thread that connects every living thing. In the quiet depths of its four bonds lies the possibility of all form, all motion, and all memory, the invisible framework upon which the music of life is written, endlessly, across the ages. 6. Biological macromolecules. Every living organism, from the smallest bacterium to the tallest tree, is composed of intricate molecular structures built from only a few repeating patterns. At the foundation of these structures lie the biological macromolecules, the large, complex compounds that carry out nearly every function required for life. They are the architecture, the machinery, and the memory of the living world. Carbohydrates, lipids, proteins, and nucleic acids together form the pillars of biological existence, each serving distinct yet interconnected purposes. They are built from simple ingredients, but their combinations yield the profound complexity of living systems. Within their shapes and interactions lies the continuity of all life, growth, motion, and the quiet preservation of information. What makes these macromolecules so remarkable is their scale and elegance. Each one consists of smaller molecular units, called monomers, that join together in long chains or intricate networks through covalent bonds. The process by which they link, known as polymerization, is itself an act of controlled energy exchange. Molecules come together, releasing water, and in that act of condensation, structure emerges. It is a universal language of assembly written in chemical syntax. This pattern of repetition allows living things to construct vast molecular frameworks while maintaining order and adaptability. In the still interior of the cell, thousands of these macromolecules interact, assembling and disassembling in constant rhythm, their behavior governed by both chemistry and the subtle requirements of life. Carbohydrates are often the first to greet us in the study of biochemistry, for they are the direct carriers of energy derived from sunlight. Through photosynthesis, plants convert carbon dioxide and water into glucose, the simplest form of carbohydrate, capturing the sun's radiant energy within its bonds. From this single molecule, an entire class of compounds arises: monosaccharides, disaccharides, and polysaccharides, each playing a role in storing or providing fuel. Glucose circulates in the blood of animals, nourishing cells with immediate energy. Starch stores that same energy in plants, patiently awaiting the moment it will be needed. Cellulose, another carbohydrate, builds the rigid walls of plant cells, lending trees and stems their strength. It is composed of the same glucose units but arranged differently, forming chains that resist breakdown. The difference between digestibility and indigestibility, between fuel and structure, lies simply in the orientation of chemical bonds, a small shift with profound consequences. If carbohydrates represent energy in motion, lipids embody energy in reserve. They are composed mainly of carbon, hydrogen, and oxygen, but unlike carbohydrates, they contain far fewer oxygen atoms. This difference gives lipids a hydrophobic nature. They repel water, forming the boundaries and barriers that define life. A lipid molecule can take many forms: oils, waxes, phospholipids, and steroids, each with its own quiet function. Fats and oils store energy compactly, holding more than twice the energy of carbohydrates per gram. Phospholipids, meanwhile, organize themselves into bilayers, creating the delicate yet resilient membranes that enclose cells. In these membranes, one end of each molecule is drawn to water while the other turns away, forming a self-assembling sheet that separates the interior of the cell from its surroundings. Steroids such as cholesterol add flexibility to these structures and act as precursors for hormones that regulate growth and metabolism. Lipids are not as visible or as active as other macromolecules, but their role is essential. They provide the shape, the insulation, and the quiet storehouses of energy that sustain every living system. Among all macromolecules, proteins are the most versatile and dynamic. They are the laborers of the cell, carrying out reactions, forming structures, and communicating signals. Each protein begins as a linear chain of amino acids, small organic molecules that contain nitrogen, carbon, hydrogen, oxygen, and sometimes sulfur. There are 20 standard amino acids, each with its own side chain that determines how it interacts with others. Once assembled, these chains fold into complex three-dimensional forms, guided by the chemical attractions between their atoms and the surrounding water. The result is a unique structure perfectly suited to its task: an enzyme that catalyzes a reaction, a fiber that provides strength, a receptor that senses a signal. This folding process, though invisible and silent, is one of the most delicate dances in nature. A single misplaced bond can cause the entire structure to collapse or lose function, as seen in diseases where proteins misfold. Yet when the process succeeds, it yields molecules of astonishing precision. Tiny machines, operating with atomic-level accuracy inside every cell. Enzymes, which are themselves proteins, deserve special attention in the world of macromolecules. They are the catalysts that make life's chemistry possible, lowering the energy required for reactions and allowing them to proceed at the gentle temperatures and pressures of living organisms. Without enzymes, even the simplest metabolic processes would take years or centuries to occur. With them, reactions happen in fractions of a second. Each enzyme recognizes its specific substrate, binds to it, and transforms it through a sequence of molecular interactions that are both predictable and profoundly efficient. The precision of these reactions reveals the quiet intelligence of chemistry, an order that requires no thought, only the intrinsic compatibility of form and function. Then there are the nucleic acids DNA and RNA, the macromolecules of memory and inheritance. Built from monomers called nucleotides, they contain three essential components: a sugar, a phosphate group, and a nitrogenous base. The sugar-phosphate backbone forms the sturdy frame of the molecule, while the bases adenine, thymine, guanine, cytosine, and uracil in RNA. It is this sequence of bases that encodes the genetic instructions for building proteins and maintaining the organism's identity. DNA, in its iconic double helix, holds this information securely, while RNA serves as the messenger, translating it into the language of life's activity. From these simple building blocks arises the continuity of generations, the ability of a single cell to divide and produce another, identical in form, yet capable of evolution. Despite their differences, all four macromolecules share a common purpose. They create stability through complexity. Each interacts with the others in a vast web of biochemical relationships. Carbohydrates attach to proteins and lipids to form glycoproteins and glycolipids, molecules that allow cells to recognize one another and communicate. Lipids and proteins combine to form membranes that regulate what enters and leaves the cell. Proteins and nucleic acids cooperate in replication and repair, ensuring that genetic information remains accurate across countless generations. In this interconnected network, no macromolecule exists in isolation. Their interactions are the very definition of life, a harmony of chemistry that builds, maintains, and renews itself with quiet precision. One of the most striking qualities of these macromolecules is their universality. The same structures appear in every living thing, from the simplest bacterium to the most complex animal. This shared molecular heritage is evidence of a single origin, a deep evolutionary connection that unites all life on Earth. The proteins that drive photosynthesis in a leaf operate through principles similar to those that power the muscles of a bird. The DNA that guides the growth of a flower uses the same code that determines human traits. In every cell, the choreography of macromolecules follows the same patterns, repeating endlessly, yet never identically. It is a cosmic refrain, the same melody played on countless instruments across the tapestry of life. In reflecting upon biological macromolecules, one senses both the power and the serenity of nature's design. They embody the perfect balance between repetition and variation, stability and change. Their existence shows that complexity can arise from simplicity, that infinite diversity can emerge from a few shared principles. Each molecule, though invisible to the naked eye, participates in the grand continuity of existence, contributing to the flow of energy, the structure of form, and the preservation of memory. When we breathe, when we grow, when we dream, these macromolecules perform their quiet work, unobserved yet unceasing. In the end, they remind us that life itself is a conversation between matter and motion, a continuous dialogue of molecules shaped by time and energy. The carbohydrates feed the cells, the lipids shelter them, the proteins make them act, and the nucleic acids remind them who they are. Together, they form the intricate chemistry of being the unseen orchestra that plays beneath our every heartbeat. Their harmony is both ancient and eternal, a reminder that even in stillness, life is always moving, always building, and always renewing itself in the patient language of molecules. Carbohydrates, sugars of life. Among the great molecules that sustain life, carbohydrates hold a special place. They are the molecules of warmth and energy, the first form of nourishment, the soft light of the sun stored in matter. Every green leaf, every seed, every fruit owes its sweetness and vitality to them. Through carbohydrates, plants capture sunlight and transform it into a form that can be shared with the rest of the living world. When we eat, the energy in our food is not new. It is ancient sunlight, trapped and preserved in carbon bonds. released once more to power our cells. Carbohydrates are, in a sense, the currency of life's economy, exchanged between organisms in a continuous cycle of giving and receiving. At the most basic level, carbohydrates are composed of carbon, hydrogen, and oxygen, usually in the ratio that forms the foundation of their name. Carbon, combined with hydrate, carbon, with water. Their simplest forms are known as monosaccharides, small molecules with names that end in oz, such as glucose, fructose, and galactose. These are the building blocks from which all larger carbohydrates are assembled. Each one follows a simple chemical pattern, yet subtle differences in structure give rise to distinct behaviors and properties. Glucose, for example, serves as the principal energy source for nearly all organisms, while fructose provides the sweetness of fruits, and galactose forms part of the sugar found in milk. The human body recognizes glucose as its preferred fuel. It flows through the bloodstream, delivering energy to muscles, nerves, and organs alike. When two monosaccharides join, they form a disaccharide, a slightly larger and more complex sugar. The most familiar of these is sucrose, the sugar that sweetens fruits and dissolves into tea, composed of one molecule of glucose and one of fructose. Lactose, the sugar of milk, pairs glucose with galactose, while maltose, formed during the breakdown of starch, consists of two glucose units linked together. Though simple in design, these sugars represent life's most accessible sources of chemical energy. When they are consumed, the body breaks them down rapidly, releasing glucose into the bloodstream. In moments of need, such as during exertion or thought, these molecules provide immediate power, fueling every motion and every spark of awareness. Beyond these simple sugars lie the polysaccharides, long chains of repeating monosaccharide units linked together through covalent bonds. Their forms vary greatly. Some are compact and easily digestible, others strong and resistant, yet they all share a common origin. The most familiar of these is starch, the storage carbohydrate of plants. Within each grain of rice or kernel of wheat lies a collection of starch molecules formed through photosynthesis, waiting to be broken down by enzymes and converted into energy. In humans and animals, a similar molecule called glycogen serves the same purpose, stored in the liver and muscles as a reserve of quick energy. When the body needs fuel, enzymes break glycogen apart, releasing glucose into the blood. This process is so precise that it can respond to the faintest shifts in energy demand, maintaining balance within the living system. Another polysaccharide, cellulose, demonstrates the strength and resilience that carbohydrates can possess. Found in the cell walls of plants, cellulose is the most abundant organic compound on Earth. Its glucose units are arranged in a pattern that forms rigid, unyielding fibers, providing structural support to stems, trunks, and leaves. Unlike starch, the bonds between the glucose molecules and cellulose are oriented in such a way that most animals cannot digest them. Humans lack the necessary enzymes to break these bonds, and so cellulose passes through our digestive system as fiber, aiding in movement but yielding no energy. Yet other organisms, such as certain bacteria, fungi, and herbivores like cows and termites, possess the enzymes required to break cellulose down, transforming what appears indigestible into nourishment. In this way, carbohydrates sustain not just energy, but ecosystems, linking plant and animal life through the balance of consumption and decay. The chemistry of carbohydrates extends far beyond their role as fuel or structure. In the complex architecture of cells, sugars often adorn the surfaces of proteins and lipids, forming glycoproteins and glycolipids that act as molecular signatures. These combinations allow cells to recognize one another, to communicate, and to cooperate in forming tissues and organs. When the immune system identifies foreign invaders, It does so by reading these sugar patterns, distinguishing the body's own cells from those that do not belong. In reproduction, fertilization, and even brain function, carbohydrates quietly play their part as identifiers and messengers. They are not only energy, but also language, a chemical form of communication written on the surface of every cell. The origin of carbohydrates lies deep in the history of life itself. Through the process of photosynthesis, plants, algae, and certain bacteria capture light energy and use it to convert carbon dioxide and water into glucose and oxygen. This process, though familiar, is one of the most profound transformations in nature. It is the point where energy from the sun enters the biological world. The glucose produced by plants serves as their own source of fuel, yet it also becomes the foundation for all other life. Herbivores consume the plants, carnivores consume the herbivores, and through this chain, the energy of sunlight moves upward through ecosystems, eventually reaching every living thing. The air we breathe and the energy that warms our bodies are both gifts of this single molecular process, repeated billions of times across the green surface of the planet. Within the human body, the management of carbohydrates is a quiet dance of precision and balance. When glucose levels rise after a meal, the pancreas releases insulin, signaling cells to absorb the sugar and convert it into glycogen, or fat, for storage. When energy runs low, another hormone, glucagon, prompts the release of glucose back into circulation. This interplay maintains the body's internal equilibrium, ensuring that energy is available when needed, but not wasted. It is a system so finely tuned that even small imbalances can have profound effects. The harmony of life depends on the delicate regulation of this simple molecule, a reminder that in biology, simplicity often conceals the deepest sophistication. The sweetness we taste in sugar is only the surface of its meaning. Carbohydrates carry within them a story that spans from the light of the sun to the beating of a heart. Each grain of starch, each fiber of cellulose, each molecule of glucose is part of a continuous exchange between life and its environment. When we eat, we are participating in that exchange, reclaiming energy that was once radiant and distant. When we breathe out carbon dioxide, we return that energy to the air, completing the cycle so that another leaf may draw it in. In this way, the flow of carbohydrates connects every living creature to the greater pulse of the planet. There is a quiet poetry in their forms. A single molecule of glucose can transform within moments burned in cellular respiration to produce energy, joined with others to create cellulose, or modified to form DNA's sugar backbone. These transitions occur without fanfare, yet they sustain the living world with perfect efficiency. The same molecule that fuels the muscles of a bird in flight may once have drifted within a green leaf, shaped by sunlight and air. This continuity gives carbohydrates a timeless quality, a sense of being both ancient and ever renewed. To contemplate carbohydrates is to recognize the generosity of nature's design. Out of carbon, hydrogen, and oxygen, the simplest of materials, arise substances capable of nourishing forests, building bodies. Every heartbeat that draws energy from glucose is a quiet echo of the sunlight that first gave it form. The sweetness of fruit, the texture of grain, the strength of wood, all are expressions of the same molecular pattern. Through carbohydrates, the universe has found a way to capture energy, to store it gently, and to release it in moments of need. They are the quiet carriers of life's light, the bridge between matter and motion, between the warmth of the sun and the breath of a living being. Lipids, the silent architects. If carbohydrates are the fuels of life, lipids are its architects, the quiet builders of barriers, the keepers of warmth, and the gentle custodians of energy. They are the molecules that shape every cell, defining its boundaries while preserving its inner world. Their work is subtle, often unseen, but without them, life's delicate order would dissolve into chaos. Lipids embody the calm of structure, the grace of balance, and the quiet endurance that allows organisms to survive from moment to moment, season to season. Their chemistry is simple, yet the beauty of their function lies in its quiet precision. At the molecular level, lipids are composed mostly of carbon, hydrogen, and oxygen, much like carbohydrates, yet their arrangement is different. And this difference gives rise to entirely new properties. They contain long hydrocarbon chains that repel water, a feature that makes them hydrophobic, a word that literally means fear of water. While carbohydrates mix readily with water, dissolving into solution, lipids turn away from it, forming layers, droplets, and films. This aversion to water is not a flaw. It is their defining characteristic, the very property that allows them to create the membranes enclosing all living cells. These membranes are not barriers of isolation but of protection, allowing the cell to maintain its own internal chemistry, distinct from the outside world. In this way, lipids provide the first and most essential requirement of life, a boundary that contains and preserves. The simplest lipids are the fats and oils, molecules designed for the storage of energy. They form when glycerol, a small alcohol, binds with 3 long fatty acid chains, producing what is known as a triglyceride. These molecules are dense with chemical energy because their hydrocarbon bonds store large amounts of potential energy that can be released through oxidation. When organisms require long-term energy reserves, they store it in the form of fat. Unlike carbohydrates, which provide quick bursts of fuel, lipids offer endurance. A single gram of fat yields more than twice the energy of a gram of sugar. In animals, fat is stored beneath the skin and around vital organs, where it also serves as insulation and cushioning, keeping the body warm and protected. The bear's winter hibernation, the migration of birds, and even the beating of a human heart during long fasting periods all depend on the patient energy held within fat molecules. The difference between fats and oils lies mainly in their degree of saturation, a subtle but important distinction in the structure of their carbon chains. Saturated fats contain no double bonds between carbon atoms, allowing the molecules to pack tightly together. This tight arrangement makes them solid at room temperature, as in butter or animal fat. Unsaturated fats, on the other hand, contain 1 or more double bonds, creating small bends in their structure that prevent tight packing. These remain liquid, flowing freely as oils. The presence of these double bonds gives unsaturated fats greater fluidity, a property that carries over into the membranes of living cells, where flexibility is vital. Even in this simple distinction, nature achieves remarkable variety, the same chemical principle, producing the solidity of fat and the softness of oil, each serving a purpose within the rhythm of life. Beyond their role as energy stores, lipids are also the architects of cellular form. The most elegant expression of this is found in the phospholipid, a molecule that embodies both attraction and repulsion. One end of the molecule, containing a phosphate group, is hydrophilic. It loves water, while the long fatty acid tails are hydrophobic. When placed in water, phospholipids align themselves spontaneously into bilayers, with their water-loving heads facing outward and their water-fearing tails tucked inward. This self-assembly creates the plasma membrane, the defining structure of every cell. Within this thin, flexible film, countless proteins float and move, carrying signals, transporting nutrients, and maintaining the cell's integrity. The membrane is both barrier and bridge, selectively allowing passage to some substances while keeping others out. It is the quiet intelligence of nature expressed in molecular form, a boundary that separates, without severing, that encloses, while still allowing exchange. Steroids, Another class of lipids add further sophistication to this picture. Their structure differs from the long chains of fats, consisting instead of 4 interlocking carbon rings. Cholesterol, perhaps the best known steroid, is often misunderstood in human health, but within cells, it performs indispensable functions. It stabilizes membranes, ensuring they remain neither too rigid nor too fluid, and it serves as the precursor for vital hormones, including estrogen, testosterone, and cortisol. These hormones, though produced in minute quantities, regulate processes as vast as metabolism, reproduction, and stress response. From the quiet foundation of lipid chemistry arises the complexity of behavior, growth, and emotion, an elegant testament to the unity between molecular structure and life's expression. Waxes, another form of lipid, provide a layer of protection for many organisms. They coat the leaves of plants, preventing excessive water loss under the heat of the sunday. On the feathers of birds and the fur of animals, they offer waterproofing and sheen, allowing the creatures to endure wet and cold conditions. Even within the human body, waxy lipids play protective roles, lining the ear canal and forming part of the outer layer of skin. Wherever they occur, they speak of endurance and preservation, the ability of living systems to resist the eroding effects of time and environment. The hydrophobic nature of lipids, which might seem an obstacle in a world so dominated by water, has instead become one of evolution's greatest advantages. In creating boundaries, lipids make individuality possible. A cell is not merely a droplet of chemicals, it is a structured entity, self-contained and self-regulating, capable of maintaining conditions distinct from those outside it. Without lipids, there could be no cells, no organisms, no continuity. The balance between water and oil, attraction and repulsion, lies at the root of all biology. It is a simple principle with infinite consequence, that life depends on separation as much as connection, on shelter as much as openness. Inside the body, lipids also play a quiet role in communication. Certain lipids act as messengers, transmitting signals between cells. Prostaglandins, for example, are lipid-based molecules that help regulate inflammation, blood flow, and the contraction of smooth muscles. They are produced only when needed, acting locally and fading quickly once their task is complete. Others, such as phosphatidylserine, participate in signaling processes within membranes, guiding the movement of ions or marking cells for renewal. Through these subtle acts of communication, lipids demonstrate that even the most seemingly inert molecules can take part in the orchestration of life's dynamic processes. When viewed on the grand scale of biology, lipids appear as agents of quiet endurance. They are the patient keepers of energy, the stabilizers of form, the mediators between fluidity and structure. They insulate, protect, and sustain. In the Arctic, layers of fat allow whales and seals to survive the freezing depths. In the desert, waxy coatings help plants conserve moisture beneath relentless sunday. Within human beings, lipids shape every cell, cushion every organ, and store the reserves that carry us through hunger and rest. Their presence is so fundamental that we rarely consider it, yet their absence would mean immediate dissolution, the loss of shape, of boundary, of life itself. There is a quiet wisdom in the design of lipids. They remind us that stability often arises from contrast, from the interplay between what mingles and what separates, what flows and what holds firm. In their gentle refusal to dissolve, lipids create the possibility of structure. In their stored energy, they promise renewal. The cell membrane, that delicate balance of water-loving and water-fearing parts, mirrors the balance required of all living things. To remain distinct yet connected, firm yet flexible, sheltered yet open to the world. In contemplating lipids, one feels a kind of stillness, the sense of something enduring and protective, something ancient. They form the invisible architecture of life, built not from rigid walls but from soft, responsive layers that breathe and move. Within their quiet chemistry lies one of nature's most profound truths, that life is sustained not only by motion and reaction, but by the patient strength of structure, by the silent architects who hold everything together, one gentle bond at a time. Proteins, the living machinery. If life could speak in the language of molecules, its words would be written in proteins. They are the artisans of the cell shaping, building, repairing, and maintaining every structure that exists within living matter. Every heartbeat, every muscle contraction, every thought and breath is the result of proteins quietly performing their duties with unimaginable precision. They are not a single kind of molecule, but a vast family, each one folded into a unique shape, each designed for a specific purpose. Within their folds lies the subtle intelligence of nature, the quiet choreography of atoms that allows life to move, to sense, to adapt, and to endure. At their core, proteins are long chains of amino acids linked together by covalent bonds known as peptide bonds. These chains are called polypeptides, and there are 20 standard amino acids that serve as their building blocks. Each amino acid has the same basic structure: a central carbon atom attached to an amino group, a carboxyl group, a hydrogen atom, and a side chain, a small chemical group that gives each amino acid its distinct personality. Some side chains attract water, others avoid it. Some are charged, others neutral. The sequence of these amino acids, arranged with mathematical precision, determines everything about the final protein's shape and function. From that simple sequence arises the diversity of life's forms, the strength of bone, the elasticity of skin, the spark of nerve impulses, and the rhythm of the heart. The making of a protein begins with the message carried in DNA. Within the quiet coils of the double helix lie instructions written in a 4-letter code, spelling out which amino acids must be linked and in what order. This information is transcribed into RNA, which then travels to the ribosome, the cell's molecular workshop. There, one by one, amino acids are brought into place, their sequence growing as the ribosome moves along the RNA strand like a careful craftsman reading a blueprint. The process is steady, methodical, and extraordinarily accurate. Even a single misplaced amino acid can alter the protein's shape, sometimes harmlessly, sometimes catastrophically. The entire process of translation is an act of precision and patience, nature's own quiet artistry, unfolding molecule by molecule. Once a protein is assembled, it begins to fold. This is one of the most mysterious and delicate transformations in biochemistry. Guided by chemical attractions and repulsions among its side chains, the linear chain of amino acids twists and bends into a 3-dimensional structure. Some sections form coils or sheets, while others loop and curve, finding the configuration of lowest energy, the shape in which the molecule is most stable. The final form may be globular, compact, and spherical like an enzyme, or elongated and fibrous like collagen or keratin. The folding process happens spontaneously, without any conscious direction, yet the results are astonishingly consistent. This is nature's quiet genius, the ability of matter to arrange itself into forms capable of thought, motion, and emotion, simply by following the laws of chemistry. The variety of proteins in living systems is nearly infinite. Some, such as enzymes, catalyze reactions, speeding them up by factors of millions. Others, like antibodies, recognize foreign substances and neutralize them. Structural proteins like collagen give tissues strength and resilience, while actin and myosin work together to generate movement in muscle fibers. Hemoglobin carries oxygen through the bloodstream, while keratin forms the protective layers of skin hair, and nails. Even within a single cell, thousands of different proteins coexist, each fulfilling its task without interference, each contributing to the harmony of the whole. The human body alone contains more than 20,000 distinct proteins, each one encoded by a gene and shaped by its specific sequence of amino acids. Among all these, enzymes stand out as the quiet catalysts of life. Without them, the chemistry of existence would proceed too slowly to sustain any organism. They lower the energy barriers of reactions, allowing them to occur quickly and efficiently under mild conditions. Each enzyme is specific, recognizing only certain substrates, the molecules upon which it acts, and binding them with exquisite precision. The fit between enzyme and substrate is often described as a lock and key, but it is more subtle than that. The enzyme may shift slightly to accommodate its partner, forming a temporary union that facilitates transformation. When the reaction is complete, the enzyme releases its product and returns to its original state, ready to act again. In this way, enzymes serve as the tireless workers of the cell, driving metabolism, replication, and repair without rest or waste. Proteins are also the instruments of communication within living systems. Many hormones are themselves proteins, or they act through receptors made of protein embedded in cell membranes. When a hormone binds to its receptor, it triggers a cascade of molecular signals inside the cell, altering metabolism or gene expression in response to changing needs. In neurons, protein channels open and close to allow ions to flow, generating the electrical impulses that underlie thought and sensation. The transmission of a single idea, the flicker of a single emotion, depends on countless proteins opening and closing in synchrony, their movements measured in milliseconds. What we perceive as consciousness emerges from the collective behavior of these silent molecular mechanisms, a harmony so subtle that it feels like magic, though it is entirely chemical in nature. The adaptability of proteins also allows life to evolve. Small changes in DNA can alter a protein sequence, leading to new shapes and new functions. Over time, these variations accumulate, giving rise to enzymes that can digest new foods, structural proteins that withstand new environments, or signaling molecules that control new behaviors. In this way, proteins record the history of life itself. The hemoglobin in our blood shares a common ancestry with that of fish and birds. The enzymes that build DNA are nearly identical in all living creatures. Each protein is a page in the long story of evolution, written in the alphabet of amino acids, refined by the slow, patient hand of time. Beyond their biological importance, proteins inspire a kind of quiet reverence for their elegance. They are the embodiment of complexity arising from simplicity, of order emerging from chance. Their forms are at once functional and beautiful, coiled helices, folded sheets, and intricate surfaces that seem sculpted with purpose. When visualized, they resemble works of art, frozen moments of motion that reveal nature's aesthetic sensibility. In their symmetry and efficiency, they remind us that life is not chaotic but deeply organized, following laws that give rise to both survival and grace. In every living cell, proteins are in constant motion, assembling, folding, unfolding, and being replaced. They do not remain static. They exist in a state of continual renewal. Old proteins are broken down, their amino acids recycled to create new ones. This turnover ensures that the machinery of life remains flexible, adaptable, and efficient. The body is never fixed but always in flux, rebuilding itself from the same molecular parts again and again. Every movement, every repair, every memory formed in the brain depends on this ceaseless molecular work. Proteins, then, are more than mere chemical structures. They are the instruments through which life expresses itself, the living machinery that allows thought, motion, and growth to occur. They bind the material and the vital, transforming simple atoms into the music of existence. Each fold, each bond, each delicate alignment of atoms speaks of patience and precision. Within their silent chemistry lies the same rhythm that animates the beating of the heart and the whisper of the wind through the leaves. To reflect on proteins is to recognize that the miracle of life is not a single event but an ongoing act of construction. Every second, countless proteins carry out their quiet duties, maintaining the order that allows us to exist. They are not conscious, yet their work gives rise to consciousness. They have no desire. Desire is made possible. In their unending motion, they embody the truth of existence itself, that life is sustained not by noise or force, but by countless acts of gentle precision, performed in silence, one bond at a time. 10. Enzymes, catalysts of calm precision. Deep within every living cell, where countless molecules drift and collide, there exists a kind of silent order, an invisible rhythm that keeps life moving forward. This rhythm is maintained by enzymes, the remarkable catalysts that make the chemistry of life possible. They are the quiet conductors of nature's orchestra, ensuring that every reaction occurs not in chaos, but in perfect harmony. Without them, the transformations that sustain life would proceed at a pace so slow that existence itself would be impossible. With them, the impossible becomes effortless. They work silently, tirelessly, and with extraordinary precision, guiding molecules into just the right positions for reactions to unfold. Enzymes are the unseen intelligence of matter not conscious, yet purposeful, turning the stillness of chemistry into the living pulse of biology. To appreciate their importance, one must first understand that every process in a living organism is a chemical reaction. Food must be broken down, energy must be released, DNA must be copied, and proteins must be assembled. All of these are reactions that require bonds to be formed or broken. Yet most of these reactions, if left to occur on their own, would take years, centuries, or even longer to complete. Life, however, cannot wait. It exists in moments, in heartbeats and breaths, in the division of cells and the flow of blood. Enzymes provide the speed that life demands. They do so not by changing the outcome of reactions, but by lowering the energy required for them to begin. They make the difficult easy, the slow swift, and the improbable inevitable. Each enzyme is a protein with a specific three-dimensional structure perfectly suited to its task. Within that structure lies a region known as the active site, a small pocket or groove precisely shaped to fit the molecules it will act upon, called substrates. When a substrate enters this site, it binds gently, like a key sliding into a lock. But the enzyme is not a rigid lock. It flexes slightly to accommodate the substrate, wrapping around it in a snug embrace. This subtle adjustment, known as induced fit, ensures perfect alignment between the reacting parts of the molecules. Once they are held in position, the enzyme facilitates the reaction, stretching certain bonds, bringing atoms closer, or providing a microenvironment where the reaction can occur with ease. When the process is complete, the products drift away, and the enzyme returns to its original form, ready to repeat the process again and again. This cycle can occur hundreds, thousands, or even millions of times per second, depending on the enzyme and the conditions. Some, like carbonic anhydrase in red blood cells, are astonishingly fast, converting carbon dioxide and water into carbonic acid at speeds nearly unimaginable, millions of reactions every second, all within the confines of a single cell. Others, like DNA polymerase, work with deliberate care, ensuring that every new strand of DNA is copied with exquisite accuracy. Whether swift or measured, each enzyme performs its task with unfailing precision, maintaining the balance of life without ever resting. The beauty of enzymes lies not only in their speed but in their selectivity. Each enzyme recognizes only specific substrates and catalyzes only a particular reaction. This specificity ensures that the chemistry of life proceeds in a controlled, orderly fashion. In the human body, thousands of different enzymes operate simultaneously, each attending to its unique role, breaking down sugars, synthesizing hormones, transmitting nerve signals, or repairing damaged DNA. They do not interfere with one another. Instead, they work in quiet cooperation, forming networks of reactions known as metabolic pathways. In these pathways, the product of one enzyme becomes the substrate for the next, creating a continuous flow of transformation. The entire process resembles an intricate assembly line, yet it operates without command, driven purely by the properties of molecules and the gentle guidance of enzymes. Enzymes are sensitive to their surroundings, responding to changes in temperature, pH, and concentration. Too much heat can cause them to unfold, losing their precise shapes and rendering them inactive. Too little warmth slows their movements, dimming the spark of metabolism. Similarly, each enzyme functions best within a narrow pH range, reflecting the environment of its natural home. The stomach's enzymes thrive in acidity, while those in the bloodstream prefer neutrality. This sensitivity underscores their delicate balance. They are both strong and fragile, capable of immense power, yet easily disrupted. In this way, enzymes mirror life itself, resilient and adaptable, but always dependent on harmony. Some enzymes require helpers, known as cofactors, or coenzymes to perform their work. These are often metal ions or small organic molecules that assist in transferring electrons or atoms during the reaction. For example, many enzymes depend on magnesium, zinc, or iron, while others rely on coenzymes derived from vitamins, such as NAD or FAD. These helpers act as silent partners, carrying energy or fragments of molecules from one reaction to another. Their cooperation adds another layer of interconnectedness to biochemistry, a reminder that no process in life stands alone. Every enzyme, every cofactor, every molecule contributes to the greater whole, part of a vast and seamless system of transformation. Enzymes also play a crucial role in regulating life's pace. Cells can control when and how enzymes act, turning them on or off as needed. Some are produced only when specific conditions arise, while others change their shape in response to molecules that bind to them. This regulation prevents waste and maintains balance. When energy is plentiful, certain enzymes slow down. When energy is scarce, others accelerate, ensuring that the organism always remains in equilibrium. The interplay of activation and inhibition forms a molecular rhythm, a breathing pattern at the level of chemistry, guiding metabolism with the same steadiness that a heartbeat guides blood. In evolution, enzymes have shaped the very course of life's possibilities. The earliest forms of life likely depended on primitive catalysts made of metal ions and simple peptides. Over billions of years, natural selection refined these rudimentary systems into the highly specialized enzymes we know today. Each step forward opened new pathways, allowing organisms to exploit new environments, digest new nutrients, or build new molecules. The enzyme that splits sugar paved the way for energy-rich metabolisms. The enzyme that copies DNA made inheritance possible. Every innovation in biology can be traced back to a molecular craftsman, an enzyme that learned, through time and adaptation, to make chemistry faster, gentler, and more precise. The quiet work of enzymes continues unceasingly in all living things. In a single moment, within your body, billions of enzymatic reactions are taking place, breaking down food, converting it to energy, assembling new cells, and maintaining the delicate balance of your internal environment. None of this requires thought or will. It happens naturally, guided by the subtle geometry of molecules and the patience of time. Each enzyme performs its task faithfully, never resting, never faltering, existing only to make life possible. If one listens closely to the concept of enzymes, there is something deeply calming in their constancy. They represent a world that moves not through force, but through cooperation, not through chaos, but through quiet order. Their precision reveals that life does not depend on miracles, but on the patient unfolding of natural laws. A cell functions not because it commands, but because its molecules understand one another through the language of shape and attraction. In that sense, enzymes are more than catalysts. They are the silent wisdom of nature made manifest in matter. To reflect on them is to glimpse the gentle intelligence of the universe itself, expressed in the smallest scales. They do not think, yet they achieve perfect purpose. They do not rest, yet they do not hurry. They work in silence, and through that silence, they sustain everything that breathes, grows, and feels. Enzymes remind us that power need not be loud, that precision can coexist with stillness, and that the true engine of life is not the dramatic spark, but the steady, unseen pulse of calm precision, the rhythm of existence itself, carried forward by molecules that never sleep. 11. Nucleic acids, the blueprint of being. In the quiet architecture of life, there exists a pair of molecules more enduring than any structure, more precious than any treasure, the nucleic acids. They are the keepers of memory, the blueprints of all living form, the written language of existence itself. Within their delicate spirals is recorded the story of every organism that has ever lived, the instructions for building bodies, minds, and generations yet to come. Nucleic acids, DNA, and RNA are not alive on their own, yet they contain within them the potential for life in its entirety. They are the bridge between chemistry and identity, the molecules through which life remembers and renews itself, age after age, in perfect silence. The structure of these molecules, though complex, is built upon simple principles. Nucleic acids are long chains of repeating units called nucleotides, each consisting of 3 components: a phosphate group, a 5-carbon sugar, and a nitrogenous base. The sugar and phosphate alternate to form the sturdy backbone of the molecule, while the bases adenine, thymine, cytosine, and guanine in DNA, or adenine, uracil, cytosine, and guanine in RNA reach inward like rungs on a twisted ladder. It is the sequence of these bases that carries the information of life, just as letters form words and words form sentences. The beauty of this system lies in its simplicity. Only 4 bases are needed to encode the infinite variety of living forms, a reminder that complexity often emerges from the humblest beginnings. Deoxyribonucleic acid, or DNA, is the more stable of the 2 nucleic acids. It exists most famously as a double helix, 2 strands coiled around one another like a gentle spiral staircase. The strands are held together by hydrogen bonds between complementary bases, adenine pairs with thymine, and cytosine pairs with guanine. This complementarity allows DNA to copy itself with remarkable fidelity. When a cell divides, the 2 strands separate, and each serves as a template for a new partner strand. The result is 2 identical copies, each containing the full set of genetic instructions. This process, replication, is one of nature's most elegant achievements, a quiet act of renewal that ensures continuity from one generation to the next. DNA resides in the nucleus of eukaryotic cells, coiled around proteins called histones, forming structures known as chromosomes. Within these chromosomes lie the genes, the specific segments of DNA that encode instructions for building proteins. Each gene is like a paragraph in the vast book of the genome, describing how to assemble a particular molecule or regulate a particular process. The order of the bases along the DNA strand determines the order of amino acids in a protein, and thus its shape and function. Though the code is universal, the same in a bacterium as in a whale, its expression creates endless diversity. From this shared molecular language arises all the variety of life on Earth, from the flutter of an insect's wing to the complexity of human thought. Ribonucleic acid, or RNA, serves as DNA's messenger and collaborator. It is usually single-stranded and more transient, designed not to store information permanently, but to carry it from one place to another. When a gene is activated, a corresponding strand of RNA is synthesized in a process called transcription. This RNA, known as messenger RNA or mRNA, leaves the nucleus and travels to the ribosome, where it guides the synthesis of proteins. Other types of RNA assist in this process. Transfer RNA, or tRNA, delivers the amino acids that will form the protein, while ribosomal RNA, or rRNA, helps assemble them in the correct order. Together, these molecules translate the static information of DNA into the dynamic structures of living cells. RNA also plays roles beyond this translation. Some RNA molecules, known as ribozymes, can catalyze chemical reactions on their own, much like enzymes. This discovery revealed that RNA is not merely a passive messenger, but a versatile and active participant in the chemistry of life. It can carry information, perform reactions, and even regulate which genes are turned on or off. Many scientists believe that life itself began in an RNA world, a time before DNA and proteins existed, when RNA molecules both stored information and catalyzed their own replication. Over billions of years, these molecules evolved greater complexity, eventually giving rise to the DNA-based life we know today. In this way, RNA may be the ancient ancestor of all biology, the molecule that first bridged the gap between chemistry and self-replication. The continuity of life depends on the extraordinary fidelity of nucleic acids. DNA replication is carried out by enzymes that check and recheck each base, ensuring that mistakes are rare. Yet occasional errors, mutations do occur, and these small changes are the raw material of evolution. Some mutations are harmful, others neutral, and a few confer advantages that allow organisms to adapt to their environments. Over time, the accumulation of these tiny variations shapes the diversity of species. Thus, within the apparent perfection of DNA's design lies room for change, for growth, for the unfolding of life's endless forms. In its balance between stability and variability, DNA reflects the rhythm of life itself, continuity, tempered by transformation. The scale of information contained within nucleic acids is staggering. A single human cell contains roughly 3 billion base pairs of DNA, organized into 23 pairs of chromosomes. If stretched end to end, the DNA from one cell would measure about 2 meters, yet it fits neatly within the microscopic space of the nucleus. In the body as a whole, with its trillions of cells, the total length of DNA would stretch beyond the sun and back many times over. And yet, all this vastness is built from a single repeating unit, the nucleotide assembled with precision and patience. The enormity of life's information is contained within the smallest of frameworks, a testament to nature's quiet efficiency. Beyond its biological function, DNA carries a kind of poetry. Its structure embodies symmetry and balance, its strands winding together in perfect proportion. Each twist of the double helix mirrors the order that runs through all of nature. The spiral of galaxies, the curl of seashells, the unfolding of ferns. Within that spiral lies both fragility and endurance. DNA can be damaged by radiation or chemicals, yet cells possess repair mechanisms that restore its integrity. Even in death, fragments of DNA can persist for thousands of years, whispering traces of the life that once was. In this persistence, one senses something timeless, the continuity of form and pattern, the memory of existence carried forward through matter itself. When we consider the role of nucleic acids in human life, the perspective deepens. Every trait we inherit, every feature of our bodies and minds, is guided by the sequence of bases passed to us from our ancestors. Yet the same molecule that determines the color of a flower or the shape of a fin also underlies the capacity for thought, art, and compassion. DNA does not know beauty or meaning, yet it creates beings capable of seeking them. In that paradox lies one of the quiet wonders of life, that from blind chemistry arises awareness, and from structure comes soul. The study of nucleic acids has transformed not only science, but humanity's understanding of itself. With the discovery of DNA's structure in the mid-20th century, the boundary between mystery and knowledge began to blur. We learned that the code of life is universal, shared by every creature that walks, swims, flies, or blooms. We learned that the differences between species are matters of sequence, not essence. This understanding has brought both humility and awe-humility in realizing how small the variations are that separate us from other beings, and awe in recognizing the unity that connects all life. In their elegance, nucleic acids teach a quiet lesson about continuity. They remind us that life is not a single spark, but a flame passed carefully from one generation to the next. The same molecules that shaped the first cell still flow within us, writing and rewriting the story of existence with every birth. They do not speak, yet they carry the oldest language in the world, a language written in 4 simple letters, yet capable of expressing everything that lives. Within their spirals lies the echo of every dawn, every heartbeat, every beginning and end. They are the blueprints of being, the silent script of creation, endlessly copying, endlessly remembering, ensuring that life, in all its delicate complexity, never forgets itself. 12. Energy and the molecule of power. Every living thing, from the smallest bacterium hidden in the soil to the largest whale moving through the deep ocean, is bound by the same quiet truth: life depends on energy. It flows through every breath, every heartbeat, and every act of growth or motion. It is invisible, yet it gives form and persistence to all that lives. Within each cell, energy moves softly, exchanged through delicate molecular gestures, transformed again and again but never lost. The molecules of life are not static. They are always at work, building and breaking, absorbing and releasing energy with the precision of an ancient rhythm. And at the center of this rhythm stands a single molecule, adenosine triphosphate, or ATP, the small yet mighty carrier of life's power. Energy, in its essence, is the capacity to do work, to move, to change, to build. In living organisms, energy manifests as chemical potential stored in bonds between atoms. When these bonds are rearranged, energy is released or absorbed. Yet life cannot rely on random bursts of power. It requires controlled, measured exchanges that can be directed toward specific tasks. The cell is not a furnace, but a finely tuned engine, where each reaction feeds the next in perfect sequence. ATP serves as the currency of this energy economy, a molecular coin that can be spent and regenerated endlessly, moving energy where it is needed and when it is needed. ATP is composed of 3 parts: a nitrogenous base called adenine, a sugar known as ribose, and 3 phosphate groups linked in a row. It is in these phosphate bonds that the molecule's energy resides. The bonds between the phosphate groups, especially the last 2, are rich in potential energy because of the repulsion between their negatively charged atoms. When one of these bonds is broken, when ATP loses a phosphate and becomes adenosine diphosphate, or ADP, energy is released. This energy is not released as heat or light, but as a gentle burst that can drive the chemical reactions of life. It powers the contraction of muscles, the synthesis of molecules, the transport of substances across membranes, and even the transmission of nerve signals. Every movement, thought, and breath depends on the breaking of these bonds, repeated billions of times within each cell every second. The beauty of ATP lies not only in its energy, but in its cycle. Once ATP has released a phosphate and transformed into ADP, It is not discarded. Instead, it is recharged, like a battery, through cellular respiration, the process by which food molecules are broken down to release energy. This energy is used to reattach a phosphate group to ADP, recreating ATP. In this way, the molecule is constantly renewed, passing through cycles of energy release and capture that never truly end. In a single human cell, millions of ATP molecules are consumed and regenerated each second. The total mass of ATP recycled in a day is greater than the mass of the human body itself. Life is therefore not a state of being but a continuous flow, a dance of creation and renewal, driven by invisible sparks of chemical energy. The process that fuels ATP regeneration begins with glucose, the simple sugar derived from food or photosynthesis. Through a series of steps known as glycolysis, glucose is gradually broken down in the cytoplasm of the cell. Each step is guided by enzymes, ensuring that the release of energy is controlled rather than explosive. The final product of glycolysis is pyruvate, a smaller molecule that carries some of the original energy of glucose. From there, in the presence of oxygen, the process continues in the mitochondria, the powerhouses of the cell. Within these organelles, pyruvate is further oxidized, and its electrons are passed along a chain of molecules in the inner membrane known as the electron transport chain. As electrons flow down this chain, their energy is used to pump protons across the membrane, creating a gradient, a reservoir of potential energy. At the end of this process stands ATP synthase, a remarkable molecular machine. It resembles a small turbine, rotating within the mitochondrial membrane as protons flow through it. With each rotation, ATP synthase joins ADP and phosphate to form a new molecule of ATP. This is one of the most beautiful examples of nature's engineering, a nanoscopic engine that turns the chemical energy of food into the molecular currency of life. The protons that move through it are like wind through a mill, and the ATP molecules it produces are the grain that sustains every living being. Each cell contains thousands of these turbines, spinning silently, tirelessly, creating the energy that allows life to persist. Not all organisms rely on oxygen for this process. Some, such as bacteria in deep-sea vents or in the anaerobic layers of soil, draw their energy from other chemical reactions, using sulfur, iron, or nitrogen compounds as substitutes. Yet even in these distant forms of metabolism, the goal remains the same: to produce ATP. It is a molecule so fundamental that it unites all life, from the simplest microbe to the most complex creature. The universality of ATP is one of the quiet wonders of biology. It speaks of a shared ancestry, a single thread of chemical continuity stretching back billions of years to the first living cell. The production and use of ATP also reveal the balance that governs all living systems. Energy cannot be stored indefinitely. It must flow. Cells produce ATP as needed, using feedback loops to ensure that supply matches demand. When energy use rises during exercise, growth, or repair, the production of ATP increases. When the need diminishes, production slows. This constant adjustment keeps the organism in equilibrium, neither overexerting nor falling into inertia. In this balance lies the wisdom of nature's design, to provide exactly enough energy for life to move forward without waste or excess. Energy, in all its forms, connects the living and the non-living. The sunlight that strikes a leaf becomes glucose through photosynthesis. The glucose becomes ATP within a cell. The ATP drives the movement of muscles, the firing of neurons, the beating of a heart. When the energy is spent, it returns to the environment as heat, radiating outward, only to be recaptured again in the endless cycle of life. Even in death, energy does not vanish. It is released back into the world, taken up by other forms of life, woven again into new beginnings. In this constant exchange, there is no waste, only transformation. When we reflect upon the role of ATP and energy in living systems, it becomes clear that life is not a static possession, but a continuous act of becoming. Every thought we think, every step we take, is powered by molecules that have been created only moments before and will soon be replaced by others. The cells of our bodies are not merely structures, they are engines of renewal, each one humming with the silent motion of ATP being spent and regained. This ceaseless exchange is the true heartbeat of existence, not the sound within our chests, but the invisible pulse of energy flowing through us, connecting us to every living thing. There is a quiet grace in this realization. The same energy that moves through the veins of a tree moves through our own bodies. The same molecular turbine that spins in the mitochondria of a leaf spins within our cells as well. Energy is the thread that ties all forms of life together, binding the smallest microbe to the greatest creature, the earliest dawn of life to the present moment. And ATP, this simple, humble molecule, is its messenger, carrying the gift of motion, warmth, and awareness from one instant to the next. In the stillness of a resting heart or the calm rhythm of breath, ATP continues its silent labor. It does not seek recognition, and it does not rest. Its bonds break and reform endlessly, weaving the continuity of life from moment to moment. Through it, the universe has found a way to sustain complexity and consciousness from mere chemistry. Energy becomes form, and form becomes thought, and thought becomes wonder. In the gentle hum of this molecular power, one can sense the profound unity of existence, the knowledge that everything that lives draws from the same well of energy, that life, in its most fundamental form, is the universe remembering how to move. 13. Metabolism, the balance of building and breaking. Inside every living organism, beneath layers of tissue and structure, beneath thought and motion, there exists a hidden harmony, a constant movement of matter and energy, known as metabolism. It is not one process, but a tapestry of countless reactions woven together with exquisite precision. Every cell is a microcosm of transformation, where molecules are built up and broken down in a continuous cycle. Metabolism is the chemistry of life in motion, the steady pulse that keeps every organism alive, awake, and adapting. It is both creation and renewal, destruction and preservation, a perfect balance of opposing forces that together sustain the miracle of existence. To understand metabolism is to see that living beings are not static things but ongoing processes. A tree is not a fixed object. It is a continuous flow of energy, drawing sunlight and minerals into its body, releasing oxygen and water back into the air. A human being, too, is a living current, atoms entering through food and air, rearranging, then leaving as new forms. Even in sleep, our cells are working, maintaining temperature, repairing tissue, and producing the molecules needed for the next day's life. The stillness of the body conceals immense activity within, a ceaseless conversation between building and breaking, synthesis and decay. These two sides of metabolism, anabolism and catabolism, define the rhythm of life. Catabolic reactions are those that break large molecules into smaller ones, releasing energy that can be harnessed for cellular work. They are acts of liberation, unlocking the energy trapped in chemical bonds. Anabolic reactions, in contrast, build complex molecules from simpler ones, using that same energy to create structure and function. They are acts of creation, shaping the raw materials of the cell into proteins, nucleic acids, and membranes. Together, these forces mirror the broader cycles of nature: decay feeding growth, energy flowing endlessly from one form to another, nothing truly lost, only transformed. Catabolism begins with the food we consume, or, in plants, with the sugars created through photosynthesis. Glucose, fats, and proteins enter the pathways of breakdown, guided by enzymes at every step. In glycolysis, glucose is split into smaller fragments, releasing a modest amount of energy. In the mitochondria, these fragments enter the citric acid cycle and the electron transport chain, where they are fully oxidized, and their energy is transferred to molecules of ATP. Carbon dioxide and water are the final products, simple, harmless substances that return to the atmosphere and environment. Catabolism, though it may sound like destruction, is in truth a cycle of renewal, freeing the energy that once came from sunlight so it can flow again through the living world. Anabolism, the counterpart of catabolism, uses this released energy to build. Within the cell, small molecules become the components of life. Amino acids join to form proteins, nucleotides assemble into DNA, and lipids merge to create membranes. Each act of synthesis requires both raw materials and energy, often in the form of ATP. When a wound heals, when muscles grow stronger through use, when a seed swells into a sprout, it is anabolism at work, the quiet construction of form and complexity from the simplicity of atoms. The harmony between these two sides of metabolism is what gives life its stability. If breakdown exceeds building, the body weakens. If building overwhelms breakdown, the system becomes inefficient and unstable. The balance between them is not fixed but responsive, shifting gently with circumstance. Guided by the needs of the moment, metabolism is regulated through a network of enzymes, hormones, and feedback signals, each one adjusting the flow of reactions in response to change. When food is abundant, enzymes favor anabolic pathways, storing energy as glycogen or fat. When fasting or exercising, catabolic pathways dominate, releasing energy from those stores. Hormones such as insulin and glucagon act as messengers, ensuring that each cell receives what it needs without excess or deprivation. This delicate interplay resembles the rhythm of breath, an inhalation of energy, followed by an exhalation of release, a perpetual exchange that keeps the organism in perfect equilibrium. The beauty of metabolism lies not only in its precision but in its unity. Despite the diversity of life, the core pathways of metabolism are the same in nearly all organisms. The same citric acid cycle turns within the mitochondria of plants, animals, and microbes alike. The same sequence of glycolytic reactions breaks down glucose in every living cell. These shared patterns reveal a deep kinship among all forms of life, a molecular heritage that stretches back to the first single-celled organism. Across the billions of years of evolution, the chemistry of metabolism has remained nearly unchanged, a universal language written in reactions instead of words. Within the cell, metabolism does not occur randomly but in organized compartments. The cytoplasm, mitochondria, and other organelles each host specific parts of these pathways, separated but connected. This spatial organization ensures that energy flows smoothly from one reaction to the next. It also allows fine control. A reaction can be sped up in one region and slowed in another, depending on the cell's needs. The mitochondria, with their folded inner membranes, act as the quiet engines of the cell, transforming nutrients into ATP through oxidative phosphorylation. Around them, the rest of the cell carries out the countless other processes of life: synthesis, repair, communication, all powered by the energy these tiny structures produce. Metabolism also connects the individual to the environment. Through eating, breathing, and movement, organisms take in energy from the world and return it in new forms. The carbon dioxide we exhale feeds the plants that later feed us. The oxygen they release fuels our cells. Even death, in the grand scheme of metabolism, is a transformation, the return of stored energy to the soil and air, where it can rejoin the cycles of life. The molecules that once formed one creature become the nourishment for another, continuing the endless circulation of matter. Nothing in nature is wasted. everything moves toward balance. In moments of rest, metabolism slows, but it never stops. Even in sleep, the body repairs and reorganizes itself, conserving energy for the coming day. In growth, metabolism accelerates, creating new cells and tissues. In illness, it changes course, redirecting energy to defense and recovery. Each of these adjustments happens automatically, the result of millions of biochemical decisions made every second, none requiring thought or command. The intelligence of life lies not in conscious choice, but in the quiet coordination of molecules, a wisdom written into the chemistry of being. When seen through this lens, metabolism becomes more than a series of reactions. It is a reflection of life's philosophy: to endure through change, to give and receive, to build while letting go. The same principles that govern metabolism also govern the natural world: forests growing and decaying, rivers flowing and evaporating, stars burning and fading. Every living organism is a small echo of the universe's larger rhythm, a microcosm of balance sustained through constant motion. To contemplate metabolism is to recognize that we are not isolated entities, but processes within a greater whole. The food that nourishes us once belonged to the earth. The breath we draw was once exhaled by trees. The atoms within us were forged in stars long before life began. In our metabolism, we carry forward the legacy of the cosmos itself. The same patterns of energy exchange that shaped galaxies, now shaping our cells. Life, in all its quiet complexity, is the continuation of that ancient flow, refined and softened through time into the gentle pulse that we call living. There is something profoundly calming in this realization. Every heartbeat, every breath, every subtle transformation within us is part of a universal conversation between energy and matter. Metabolism, in its steady balance of building and breaking, reminds us that destruction and creation are not opposites but partners. From one comes the other, and through their endless exchange, life remains whole. To live is to participate in that balance, to become a fleeting but essential moment in the ceaseless chemistry of the universe, a small flame kindled from ancient fires, burning quietly, beautifully, and always in harmony with the rhythm that began long before we were born, and will continue long after we are gone. Cellular respiration, the dance of energy. Deep within every living cell, in a realm invisible to the naked eye, there unfolds an endless choreography, a graceful dance of molecules that sustains life itself. This dance is known as cellular respiration, the process by which cells extract energy from nutrients and transform it into a usable form. It is the quiet heartbeat of existence, the rhythm that keeps muscles moving, nerves firing, and thoughts forming. While it may appear mechanical when described in chemical terms, in truth, it is a masterpiece of natural harmony, an exchange of energy so balanced and so refined that every breath we take, every movement we make, depends upon its perfection. At its essence, cellular respiration is the process of converting chemical energy stored in food into adenosine triphosphate, or ATP, the molecule of power that fuels nearly all biological activity. The story begins with glucose, the simple sugar produced by photosynthesis or absorbed from food. Within each molecule of glucose lies the energy of sunlight, captured and stored by plants, a gift passed from the green world to the animal kingdom. To release that energy, the cell must dismantle the glucose molecule step by step, harvesting its stored potential without waste or chaos. The elegance of respiration lies in this gradual release, a slow, measured unraveling that gives life its steady power. The first act of this molecular dance occurs in the cytoplasm, the fluid space that fills the cell. Here, through a sequence of reactions known as glycolysis, glucose is divided into two smaller molecules called pyruvate. This process requires no oxygen and yields only a small amount of ATP. but it is enough to spark the continuation of the cycle. The cell invests a little energy to begin, but the return is greater, a small profit that keeps the system in motion. Along with ATP, glycolysis produces high-energy electrons carried away by special molecules like NADH, which will later deliver them to the next stages of respiration. Even in this early step, one can sense the precision of life's design. Nothing wasted, everything prepared for what comes next. If oxygen is available, the pyruvate molecules enter the mitochondria, the tiny organelles, often called the powerhouses of the cell. These structures, with their folded inner membranes, are where the deeper phases of respiration occur. In the mitochondrial matrix, pyruvate is transformed into acetyl coenzyme A, a molecule that enters the citric acid cycle, also known as the Krebs cycle. Here begins the true artistry of biochemical balance. As acetyl groups are broken down, carbon dioxide is released as a byproduct, and more electrons are transferred to carrier molecules such as NADH and FADH. The cycle turns continuously, like a wheel, generating both energy and the materials needed for other cellular processes. Each turn of this wheel is small and quiet, yet together they create the foundation of life's energy flow. From the citric acid cycle, the electrons gathered by NADH and FADH are delivered to the inner membrane of the mitochondrion, where they enter the electron transport chain. This is the final and most intricate movement of the dance. Here, electrons are passed along a series of protein complexes, each step releasing a small amount of energy. With each transfer, protons are pumped across the membrane, creating an electrochemical gradient, a reservoir of potential energy. It is this gradient that drives the synthesis of ATP, as protons flow back through an enzyme called ATP synthase, spinning it like a tiny turbine. Each rotation joins ADP and phosphate to form ATP, the molecule of life's motion. The entire process is astonishingly efficient, producing up to 30 or more molecules of ATP from a single glucose molecule. Oxygen, the gas we breathe, plays a quiet but vital role at the end of this chain. It serves as the final electron acceptor, combining with electrons and protons to form water. Without oxygen, the flow of electrons would halt, the chain would stall, and energy production would cease. This is why oxygen is so essential to complex life, not because it sustains breath itself, but because it completes the circuit of energy. The air we inhale becomes part of a molecular process that powers every cell, and the carbon dioxide we exhale is the spent remnant of that exchange. Every breath is, in truth, a partnership between body and world, an act of quiet cooperation between chemistry and atmosphere. In the absence of oxygen, some organisms and cells resort to a simpler form of respiration called fermentation. In this process, pyruvate is converted into other products, such as lactic acid or ethanol, allowing glycolysis to continue even without the electron transport chain. The yield of energy is small, but for certain microbes, or for our own muscle cells, during intense exertion, it is enough to survive. When oxygen returns, normal respiration resumes, clearing away the lactic acid that causes fatigue. This adaptability illustrates the resilience of life, its ability to adjust its dance to whatever rhythm the environment provides. Cellular respiration is not only a process of energy release, but also a guardian of balance. It ensures that the cell's energy supply matches its needs, neither overproducing nor falling short. Enzymes regulate every step, responding to signals that reflect the cell's condition. When energy levels are high, respiration slows. When energy is low, it accelerates. This self-regulation mirrors the broader cycles of nature, a constant adjustment that maintains harmony. The heat generated by respiration helps warm the body, especially in animals, turning chemical transformation into gentle life-sustaining warmth. Even in rest, the quiet burning of molecules continues, a hidden fire that never goes out. When seen in its entirety, cellular respiration reveals more than just a set of chemical reactions. It is a story of flow of electrons, atoms, and energy moving seamlessly through matter. It connects the living to the non-living, linking sunlight, air, and earth in one unbroken circle. The energy released by respiration once came from the sun, captured by plants through photosynthesis. Through food, it enters animals and humans, powering their motion and thought. When life ends, the energy returns to the environment to be taken up again by new forms of life. In this way, respiration is not simply a mechanism of survival, but a thread that ties every organism to the whole of the planet. There is something profoundly peaceful about this realization that every breath we take is an act of participation in an ancient rhythm. The air we inhale may once have nourished forests, oceans, or creatures long vanished from the earth. The glucose we metabolize carries the energy of sunlight that fell upon leaves months or even years ago. Through respiration, we are reminded that nothing in life exists alone. Each of us is part of the same grand cycle, drawing in what others have released, giving back what others will one day use. It is a quiet communion, as steady and sacred as the heartbeat itself. To reflect on cellular respiration is to glimpse the inner stillness of life's motion, beneath the noise of the world, beneath the beating of hearts and the flutter of leaves. The same silent chemistry unfolds, electrons moving along their invisible paths, protons flowing through their channels, energy shifting from one form to another. It is a dance, older than time, choreographed by the laws of nature, yet performed endlessly by living beings. Through it, the earth breathes, and we breathe with it. Each exhalation, each release of energy, is a note in the ongoing song of existence, a reminder that life, though made of countless reactions, is ultimately one continuous and graceful dance of energy. 15. Photosynthesis. The chemistry of light. There is a kind of quiet magic that unfolds each morning when sunlight touches the surface of a leaf. Invisible to the eye, an ancient process awakens the transformation of light into life. Photosynthesis is the great alchemy of the living world, the bridge between the energy of the sun and the nourishment of all that grows and breathes. It is the gentle conversation between light and matter, between the sky and the soil. Within its calm precision lies the foundation of every forest, every ocean, every breath of air that fills the lungs of living things. It is, in essence, the art of turning sunlight into the fabric of existence. At the heart of photosynthesis is a simple idea, yet one of infinite consequence. Light energy is captured and converted into chemical energy, stored in molecules like glucose. This process takes place in the chloroplasts, tiny green organelles found within plant cells and in certain algae and bacteria. Inside each chloroplast are membranes known as thylakoids, arranged in stacks like coins in a column. Embedded within these membranes are pigments, most notably chlorophyll, that absorb light and initiate the reactions that sustain the living world. It is this chlorophyll that gives plants their green hue, reflecting the color of balance and growth, while absorbing the red and blue wavelengths that fuel their work. When sunlight strikes a leaf, photons, tiny packets of light energy, are absorbed by chlorophyll molecules. This energy excites electrons within the pigment, raising them to higher energy levels. These energized electrons are then passed along a chain of proteins within the thylakoid membrane, known as the electron transport chain. As they move through this chain, their energy is gradually released and used to pump protons across the membrane, creating a gradient much like the one found in the mitochondria during respiration. This stored potential energy drives the synthesis of ATP, while other molecules, such as NADPH, carry high-energy electrons needed for the next stage of photosynthesis. In this way, sunlight is transformed into chemical energy, captured, contained, and made ready to build life's materials. But light alone is not enough. The second part of photosynthesis, known as the Calvin cycle, takes place in the fluid-filled space of the chloroplast called the stroma. Here, the energy stored in ATP and NADPH is used to fix carbon dioxide, the gas we exhale, into organic molecules. Through a series of enzyme-guided reactions, carbon dioxide is combined with a simple 5-carbon compound to form glucose and other carbohydrates. The cycle turns again and again, building energy-rich molecules that will serve as the foundation of life's food web. The oxygen released during this process is a byproduct, a quiet gift that fills the atmosphere and sustains respiration in animals and humans alike. Every breath we take is, therefore, a continuation of the conversation that began in the chloroplast, a return of the favor given by the leaves and the sea. The balance between photosynthesis and respiration is one of the most harmonious cycles in nature. Plants and algae capture energy from sunlight and produce oxygen, while animals consume that oxygen and release carbon dioxide. The waste of one becomes the resource of the other, an exchange so perfect that it has persisted unchanged for billions of years. Together, these processes create the stable conditions that make life possible: the breathable air, the flowing energy, the fertile ground upon which all living systems depend. It is a partnership without words, a relationship defined by quiet reciprocity. Chlorophyll is not the only pigment that participates in this dance of light. Carotenoids and phycobilins absorb wavelengths of light that chlorophyll cannot, extending the range of colors that can be used for energy capture. These pigments also protect the plant from excessive sunlight, dissipating surplus energy as heat to prevent damage. In autumn, when chlorophyll fades, the other pigments remain, revealing the yellows, oranges, and reds that have been present all along. The changing of the leaves is thus not the end of photosynthesis, but its graceful pause, a reminder of the cycles that govern all living things, the alternation between activity and rest. On a molecular level, the elegance of photosynthesis lies in its efficiency. It converts less than 1% of the sunlight that reaches the Earth into chemical energy. Yet this is enough to sustain every living thing on the planet. Each second, billions of tons of carbon dioxide are fixed into organic molecules by green plants, while oxygen streams back into the air. This vast exchange occurs silently and continuously, from the surface of the oceans to the tops of mountains. It requires no conscious effort, no machinery beyond the molecular, yet it shapes the fate of the planet more profoundly than any human invention. In the stillness of a forest or the shimmer of an algae bloom, the quiet labor of photosynthesis carries on, unnoticed yet indispensable. The origins of this process stretch far back in time, to the earliest days of life on Earth. Ancient cyanobacteria, living in the planet's primordial oceans, were the first organisms to develop the ability to harness light energy. Their activity slowly transformed the atmosphere, filling it with oxygen and paving the way for complex life to evolve. The world before photosynthesis was a different place, a dim, anaerobic planet with no blue sky, no rust-red soil, no breath. Over millions of years, the invisible work of these microscopic organisms reshaped the planet's chemistry and climate. The oxygen they produced allowed for the emergence of respiration, which in turn gave rise to movement, thought, and the boundless diversity of life we know today. The green of a leaf, then, is not just a color, it is a symbol of an ancient triumph, the memory of light captured and carried forward through time. Photosynthesis does not occur only in the world of plants. It also takes place within countless forms of microscopic life, algae, cyanobacteria, and even certain protists that float in oceans, rivers, and lakes. These tiny organisms, though individually small, produce more than half of the world's oxygen. They form the base of marine food webs, supporting fish, whales, and entire ecosystems. Beneath the waves, sunlight filters through the water, and there, too, the chemistry of light continues quiet, rhythmic, eternal. It is humbling to realize that the breath of every creature depends as much on these invisible drifters as on the forests of the land. In reflecting on photosynthesis, one senses the deep symmetry of existence. It begins with light, pure energy falling from the sun across vast distances, and ends with the growth of leaves, the flight of birds, the beating of hearts. Through photosynthesis, the earth itself becomes a living being, drawing energy from the cosmos and turning it into form and motion. The cycle of day and night, of light and shadow, becomes the rhythm of life's renewal. Even in darkness, the stored energy of sunlight sustains motion and thought, a reminder that life is never truly separate from the stars. There is a quiet wisdom in the simplicity of this process. A leaf does not strive or compete, it simply opens itself to the light, allowing energy to flow through it and into the world. The same sunlight that warms our skin also fills the veins of trees and grasses, shaping the air we breathe. In every green blade, in every whispering forest, the chemistry of light continues endlessly, patiently, gracefully. Photosynthesis is not only a process, it is a meditation on the nature of life itself, on balance, generosity, and the endless giving of energy from one form to another. As evening falls and the sunlight fades, the leaves rest, but the energy they captured remains, flowing through roots, stems, and seeds, moving outward into the vast web of life. In that gentle cycle, the light of the day becoming the breath of night, the quiet purpose of photosynthesis endures. It is the foundation of nourishment, the beginning of every heartbeat, the reason the planet glows with color and motion. It is the chemistry of light made manifest, the song of the sun translated into the language of life, playing softly, eternally, in every green thing that grows beneath the sky. The flow of matter, cycles of nature. In the great stillness of the earth, beneath the rising and falling of seasons, beneath the shifting winds and the silent movement of oceans, there flows a constant exchange, a ceaseless recycling of matter that sustains all life. Nothing in nature is ever truly lost or wasted. Every atom that composes a living being has been part of countless forms before drifting once as air, flowing as water, embedded in rock or soil, or residing in another living creature. The flow of matter through the natural world is one of the most profound expressions of balance, a reminder that existence itself depends not on endless creation, but on continual renewal. In this quiet circulation, the planet breathes, feeding itself through cycles that have turned unchanged for billions of years. Matter moves through nature in loops rather than lines, circulating through ecosystems in patterns of astonishing elegance. The molecules that form a leaf will one day fall to the soil and nourish new growth. The carbon exhaled by an animal becomes the raw material for a plant's photosynthesis. The water that evaporates from an ocean returns as rain upon distant mountains, flowing again through rivers and roots. Life is not separate from these cycles, but a participant within them, a momentary gathering of atoms, a brief pause in their endless motion. Even as we live and breathe, the elements that form our bodies are moving through us, never static, never wholly our own. Among the many cycles that govern the earth, the carbon cycle stands as one of the most vital. Carbon is the backbone of organic chemistry, the framework upon which life is built. In the air, it exists as carbon dioxide, a colorless gas drawn into the leaves of plants during photosynthesis. There, through the gentle power of sunlight, it is woven into sugars and starches, the molecules that serve as food for nearly every organism. When plants are consumed by animals, this carbon moves onward, becoming part of muscles, bones, and cells. When those animals breathe, decay, or burn, Carbon returns to the atmosphere, completing the cycle. It is an exchange so ancient and balanced that even the air we exhale carries the signature of stars that long ago gave birth to carbon atoms in their fiery cores. In the soil and oceans, carbon finds other resting places. Some of it becomes trapped in the shells of marine organisms, eventually forming limestone and other rocks. Some is buried deep underground, compressed over eons into coal, oil, and natural gas. When humans burn these fuels, carbon returns once more to the air, rejoining the cycle that first gave it purpose. Every candle's flame, every exhaled breath, every rustling forest, all are part of this single story of carbon's eternal journey, changing form but never disappearing. Just as carbon circulates through living systems, so too does water, the element that makes life fluid and flexible. The water cycle is a continuous exchange between sky, land, and sea. The sun's warmth draws water upward from oceans, lakes, and leaves, turning it into invisible vapor. As it rises and cools, it condenses into clouds, then falls again as rain or snow, returning to the ground. Some seeps into the soil, joining underground aquifers that sustain forests and fields through dry seasons. Eventually, all water returns to the sea, completing its timeless circuit. Every drop that falls upon the earth has already traveled this journey countless times, perhaps once coursing through the veins of a dinosaur, perhaps drifting as mist over an ancient forest long vanished. The nitrogen cycle, though less visible, is equally essential to life's continuity. Nitrogen forms the basis of amino acids and nucleic acids, the building blocks of proteins and DNA. Yet the nitrogen in the atmosphere, abundant though it is, cannot be used directly by most living things. Instead, it must be transformed. Certain bacteria possess the remarkable ability to fix atmospheric nitrogen, converting it into compounds like ammonia and nitrates that plants can absorb. Through roots, nitrogen enters the web of life, nourishing the growth of leaves and tissues. When animals consume plants, nitrogen becomes part of their bodies, eventually returning to the soil through waste or decay. Other bacteria complete the cycle, releasing nitrogen back into the atmosphere, ready to begin again. In this unseen exchange, microscopic life ensures that the world remains fertile, bridging the gap between air and earth. Phosphorus, another vital element, moves more slowly through its own cycle. Unlike carbon and nitrogen, it does not enter the atmosphere in significant amounts. Instead, it is released from rocks through the gradual process of weathering, carried by water into the soil, where plants absorb it. From there, it passes through animals and decomposers, eventually returning to sediment or the ocean floor. Though its journey is measured in millennia, phosphorus is indispensable. It forms the backbone of DNA and the energy molecules that power every cell. In its patient movement, it teaches us that not all cycles are quick. Some turn with the slowness of stone, shaping the foundation of life through the quiet work of time. Every cycle, carbon, water, nitrogen, phosphorus, and others weaves into the grand tapestry of ecosystems. Together they create balance, ensuring that no resource is ever wholly lost, that every act of consumption is followed by renewal. Even death, often perceived as an ending, is merely another beginning within these cycles. The fallen leaf nourishes the soil that feeds new roots. The decaying animal enriches the earth that sustains future generations. Decomposers, the unseen recyclers of life, break down complex molecules into simpler forms, returning nutrients to the world. Through them, the boundary between life and death dissolves, and the continuity of matter is preserved. When we look at nature through this lens, it becomes clear that everything is connected through the flow of matter. The air we breathe was once exhaled by ancient trees. The water we drink once coursed through prehistoric rivers, The calcium in our bones may have once formed the shell of a marine creature. Life is not built from new materials, but from those endlessly reused, shared, and reshaped. This realization softens the distinction between self and world, for what we call us is, in truth, a momentary arrangement of elements borrowed from the earth, destined to return to it. These cycles also reveal the wisdom of balance. In untouched ecosystems, the flow of matter maintains harmony without interference. Forests renew themselves through the fall of leaves and the growth of saplings. Oceans regulate carbon through the rise and fall of plankton blooms. Even deserts find equilibrium through rare rains and resilient life. Disturbances occur when these cycles are broken or accelerated, when matter is extracted faster than it can be returned. Yet nature, with its quiet patience, always seeks restoration. Given time, it finds its balance again, mending what was disrupted through the same principles that have guided it for eons. In the stillness of reflection, one may sense how these cycles mirror our own lives. We, too, are part of a rhythm of giving and receiving, taking sustenance from the world, transforming it, and returning it again. Every breath we draw participates in a vast conversation with the trees. Every meal connects us to the soil and the sunday. Even our thoughts, born of chemical energy, are the latest expression of atoms that once belonged to stars. The flow of matter is, in this sense, the poetry of the universe, a story written not in words, but in elements, repeated endlessly in new and beautiful forms. To contemplate the cycles of nature is to see life not as a possession but as a passage. We are custodians of the materials that pass through us, caretakers of a world that renews itself through our participation. The same carbon that fuels our breath, the same water that flows in our blood, will one day rise again, in other forms, as rain, as soil, as leaf, as life. The earth does not end and begin with us. We are part of its continuous flow, each of us a momentary expression of a timeless cycle. In recognizing this, we find peace, the understanding that nothing is ever truly lost, that all things return, and that the dance of matter, in its endless grace, is the quiet heartbeat of the living world. Shurtsi, the cell, a universe within every living being, hidden beyond the limits of the eye. There exists a world of astonishing complexity, a universe in miniature, where structure and motion, order and chaos, coexist in perfect balance. This is the cell, the smallest unit of life, and yet also one of the most profound. It is at once a factory, a community, and a living being in its own right. Inside it, molecules dance in endless rhythm, guided by unseen laws that give rise to all that breathes and grows. Each cell, though microscopic, is a complete world unto itself, capable of sensing, responding, reproducing, and maintaining its own harmony. When we gaze upon the vastness of life, from forests to oceans, to the beating of a human heart, it is humbling to remember that it all begins here, in the quiet intricacy of a single cell. The cell is both simple in concept and infinite in detail. At its most basic, it is a small compartment enclosed by a membrane, separating the inner environment from the outer world. Yet, within that boundary lies an organized realm of astonishing activity. The cell's membrane, far from being a mere barrier, is a dynamic interface selectively permeable, allowing the exchange of nutrients, gases, and signals while maintaining internal stability. Through this thin layer of molecules, the cell negotiates its existence, drawing in what it needs, releasing what it no longer requires, and communicating with the world beyond. It is through this delicate membrane that life maintains its balance, a quiet act of separation that allows individuality without isolation. Inside the cell, the cytoplasm fills the space like a gentle sea, a fluid medium in which countless reactions occur. Floating within it are the organelles, each with a distinct role, each contributing to the harmony of the whole. In the nucleus, the genetic material rests, coiled into chromosomes that contain the instructions for life. These threads of DNA are not inert. They are living archives, constantly read and rewritten, guiding the cell's every function. When the cell divides, the DNA is copied with extraordinary accuracy, ensuring that the story of life continues unbroken. It is here that memory resides, not the kind that recalls a moment or thought, but the deep, ancient memory of form and function, passed down through generations of cells since the dawn of life. Beyond the nucleus, the mitochondria serve as the cell's powerhouses, converting the energy of food into ATP, the molecule that fuels all activity. Their inner membranes are folded into intricate patterns, maximizing the surface area for the reactions of respiration. Interestingly, mitochondria carry their own DNA, a remnant of their ancient origins, as free-living bacteria that once entered into partnership with early cells. This union, known as endosymbiosis, marked a turning point in evolution, a moment when two independent forms of life merged to create something greater. Even now, every breath we take echoes that ancient alliance, as the mitochondria within us continue their patient labor, transforming oxygen and nutrients into the quiet fire of energy. In plant cells, another set of organelles carries out an equally profound task: the chloroplasts, where photosynthesis takes place. Like mitochondria, chloroplasts have their own DNA and descended from ancient organisms, in this case, cyanobacteria, that could harness sunlight. Within their green membranes, photons of light are captured and transformed into chemical energy, sustaining not only the plant, but ultimately all life that depends upon it. Each chloroplast is a small sun within the cell, gathering the warmth and brilliance of the cosmos and turning it into matter, into nourishment, into life itself. Other organelles contribute their own silent efforts to the cell's operation. The endoplasmic reticulum, a network of folded membranes, serves as a site of synthesis and transport. Its smooth regions assemble lipids and hormones, while the rough regions, dotted with ribosomes, manufacture proteins. The Golgi apparatus then receives these proteins, modifying and packaging them for delivery to their destinations. Vesicles bud off and move like small boats through the cytoplasm, carrying their precious cargo to the membrane or other parts of the cell. Lysosomes, filled with enzymes, act as recyclers, breaking down old or damaged materials so that their components may be used again. Every part of the cell is both independent and interdependent, each performing its role in service of the greater whole. This organization is not static, but alive with motion. Molecules diffuse through the cytoplasm, guided by gradients and signals. Proteins fold and unfold. Enzymes bind and release. Membranes flow and reshape themselves. Even the cytoskeleton, the network of filaments that gives the cell its structure, is constantly rearranging, allowing the cell to move, divide, and change shape. Under a microscope, this inner life resembles a vast city seen from above at night, lights flickering, pathways shifting, energy moving ceaselessly through every corridor. Yet all of it occurs without noise or confusion, governed by the quiet logic of chemistry and the steady order of time. Communication is another marvel of the cell. Through chemical signals, cells speak to one another across tissues and distances, coordinating growth, repair, and response. Hormones, neurotransmitters, and electrical impulses all serve as messages, carried by molecules that bind to receptors like keys, fitting into locks. A single signal can set off a cascade of reactions within the cell, turning genes on or off, directing resources to where they are needed, or initiating movement. In multicellular organisms, this communication gives rise to astonishing complexity. Trillions of cells working in harmony, each performing its own task, while remaining attuned to the needs of the body as a whole. The beating of a heart, the healing of a wound, the opening of a flower, all arise from this unseen dialogue between cells. Despite their intricacy, all cells share certain features that link them across the vast diversity of life. Whether in a bacterium floating in the ocean or a neuron in the human brain, the same basic principles hold true: a boundary, a flow of energy, a system of communication, and a cycle of renewal. This unity speaks to a shared origin, a single ancestral cell from which all others have descended. Over time, evolution has diversified their forms, some large and complex, others small and simple, yet the essence remains unchanged. Within each cell, one can trace the continuity of life itself, the unbroken chain that connects every organism that has ever existed. In many ways, the cell reflects the structure of the universe itself. It is a cosmos enclosed in a membrane, filled with forces, motion, and purpose. The nucleus stands like a sun at its center, radiating information outward, while organelles orbit and interact like planets in a solar system. The cytoplasm hums with the same kind of dynamic energy that fills galaxies with motion. Just as stars are born, burn, and collapse, so too do cells arise, grow, divide, and die, only to give rise to new generations. To look into a cell is to look into the fabric of existence, the same principles of balance, connection, and transformation echoed at every scale of reality. When one contemplates the cell, a sense of quiet wonder naturally arises. Every sensation we experience, every movement we make, is the product of countless cells working together, each performing its part without pause. They ask for nothing, yet they give everything. They are born, they live, they die, and through their endless cycle, the greater organism endures. To live is to host an entire universe within, a symphony of molecular life, performing ceaselessly beneath our awareness. And so the cell, though unseen, holds the essence of being. It teaches that life is not a single entity but a collaboration, a harmony of countless parts acting as one. Each cell carries within it the history of the earth and the potential of the future, written in the language of chemistry and time. When we breathe, move, or dream, we are participating in their work, the quiet, patient labor that has sustained the planet since the beginning. The cell is not merely a structure, it is a miracle of continuity, the smallest reflection of the cosmos itself, alive with the same quiet intelligence that moves through stars, oceans, and hearts alike. 18. The origin of life. From chemistry to consciousness. Long before there were forests, rivers, or even oceans filled with creatures, the earth was silent and raw, a place of storms and stone, of lightning and volcanic fire. The air was heavy with gases. The seas were dark and restless. And yet within this seeming chaos, something extraordinary was beginning. Somewhere in the depths of that ancient ocean, the first whispers of life stirred. There was no sudden spark, no single miraculous moment, but rather a long unfolding, a gradual awakening of order within disorder, of pattern within chaos. From the simple interactions of atoms and molecules, the first living systems emerged, fragile and tentative, yet bound for infinite possibility. The story of life's origin is not one of magic but of natural beauty, written in the quiet logic of chemistry. In those early days, Earth's surface was rich with elements: carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus, the same elements that form every living thing today. Energy from sunlight, lightning, and volcanic heat flowed freely, driving reactions among the gases in the atmosphere and the minerals in the sea. Over time, simple molecules combined into more complex ones: amino acids, nucleotides, and sugars. These were the alphabet of life, the raw materials from which cells and organisms would one day be built. They drifted through the waters, reacting, breaking apart, and forming again, until eventually some found stability, some began to replicate, and from that replication arose the first fragile continuity, the beginning of evolution itself. One of the great mysteries of science lies in how these molecules organize themselves into systems capable of self-replication and metabolism. Theories abound, each offering a glimpse into this ancient process. Some suggest that life began near hydrothermal vents deep in the ocean, where hot, mineral-rich water provided both energy and structure. In these vents, natural compartments formed within porous rocks, creating sheltered environments where molecules could concentrate and interact. Others propose that life's first ingredients arrived from space, carried by comets or meteorites that delivered organic compounds to Earth's surface. In truth, the boundary between Earth and the cosmos may not have been as sharp as we imagine, for the same elements that formed the stars also formed us. At some point, simple molecules began to form chains long, repeating sequences capable of storing information. RNA, a molecule still used by life today, may have been the first of these. It has the remarkable ability to act both as a carrier of information and as a catalyst, driving chemical reactions. In what scientists call the RNA world, these molecules may have replicated themselves, using the energy of their surroundings to assemble copies. Over time, variations arose, some more stable, some more efficient, and natural selection began to shape chemistry into something new, life that could evolve. From there, cooperation emerged. Molecules began to assist one another, forming networks of reactions that supported mutual survival. It was in these small alliances that metabolism was born, the first whispers of the biological harmony that still sustains us today. The next great step was the creation of a boundary, a membrane to separate the living from the non-living, the inner from the outer. Fatty acids and other lipids, abundant in the ancient seas, naturally formed spheres in water, with their hydrophobic tails turned inward and their hydrophilic heads facing outward. These primitive vesicles, though simple, could enclose molecules, creating tiny pockets of order within the surrounding chaos. Inside these membranes, chemical reactions became more efficient, more self-contained. Molecules could replicate without being lost, and the first true protocells took shape. They were not yet alive in the modern sense, but they had crossed a threshold. They could grow, divide, and evolve. The spark of persistence had been kindled, and the story of life had begun. Over countless generations of chemical change, these protocells grew more complex. RNA gave rise to DNA, a more stable molecule for storing genetic information, while proteins emerged as the versatile tools of biochemistry. The first cells capable of metabolism and replication appeared, and the long, slow process of evolution began to sculpt the diversity of life. For millions of years, these early cells lived in the ocean, feeding on the chemical richness of their surroundings. Some learned to capture light, developing the first forms of photosynthesis. This transformation, the ability to turn sunlight into energy, changed the world forever. Oxygen began to fill the atmosphere, and with it came new possibilities. From the calm depths of ancient seas arose the first great expansion of life, an unfolding that continues to this day. The beauty of life's origin lies not in its improbability, but in its inevitability. Given the right conditions, energy, water, and time matter seems to organize itself into ever more intricate forms. The same physical laws that shape galaxies and stars also shape the molecules of life. Hydrogen bonds, van der Waals forces, and the geometry of carbon atoms guided the assembly of molecules long before consciousness ever emerged to ponder it. Life is not separate from the universe, but an expression of it, a natural consequence of its tendency toward complexity and pattern. From dust and fire came structure and memory, From molecules came movement and awareness. Even now, the early history of life echoes in every living cell. The genetic code we carry is written in the same alphabet that existed billions of years ago. The membranes that protect our cells are built from the same kinds of lipids that once formed the first protocells. The enzymes that sustain our metabolism are the descendants of ancient catalysts that once worked in the shadows of hydrothermal vents. Every breath we take, every heartbeat, every thought is part of the same unbroken thread that began in those early waters. The origin of life is not a distant event, frozen in time. It is an ongoing process, renewed in every cell that divides, in every organism that grows and adapts. And then, somewhere along that immense timeline, life began to look outward. From simple reactions came sensitivity. From sensitivity came awareness. Nerve cells evolved, linking together into networks that could respond to the world in ever more complex ways. Eyes formed to see the light that had always been there, ears to hear the vibrations of air and water, minds to reflect upon existence itself. The atoms that had once swirled in the dark oceans learned to dream of the stars. Consciousness, too, is part of this same continuum, not a separate creation, but the latest flowering of a process that began with chemistry, learning to persist. When we trace our origins back to those first molecules, a profound humility arises. The boundaries between the living and the non-living begin to blur, replaced by a deeper understanding that all things are part of one continuous story. The rocks, the seas, the air, and the creatures that inhabit them are all expressions of the same cosmic fabric, shaped by time and energy into infinite variation. Life is the universe becoming aware of itself, a way for matter to reflect on its own existence. Through us, the earth contemplates its own beauty. Through our thoughts, the cosmos remembers its origins. The origin of life, then, is not merely a question of beginnings, but of continuity. It is the realization that life has always been part of the universe's unfolding, that every element within us once belonged to something vast and ancient. We are made of the same carbon forged in stars, the same water that has cycled through oceans since the dawn of time, the same molecules that first learned to replicate in the quiet depths of the sea. When we breathe, we continue the work that began billions of years ago, the ceaseless exchange between energy and matter, between chemistry and consciousness. To ponder this is to feel a kind of calm wonder. The story of life is not a tale of sudden miracles, but of gentle persistence. It is the story of atoms finding companionship, of molecules learning to remember, of cells learning to feel. It is the same story written again and again in every living thing that has ever existed. From chemistry to consciousness, from ocean to mind, life has been unfolding quietly, patiently, beautifully, a single thread of becoming woven through time, carrying the memory of stars within every heartbeat and the promise of eternity within every breath. 19. Evolution, the endless adaptation. Across the vast sweep of time, life has never stood still. It has shifted, changed, and adapted, endlessly reshaping itself in response to the turning of the earth, the changing of climates, and the silent pressures of survival. This grand transformation, known as evolution, is both the history and the poetry of life. It is the process through which simple forms have given rise to complexity, through which the smallest cell has become the forest, the ocean, the bird, and the human being. Evolution is not a march toward perfection, nor a design imposed from above. It is the patient unfolding of possibility, the continuous conversation between life and environment, guided by chance yet refined by necessity. When we speak of evolution, we are speaking of time so deep that it humbles imagination. The story begins not in centuries or millennia, but in billions of years, a span of time during which oceans have risen and vanished, mountains have formed and eroded, and entire species have come and gone like brief notes in a cosmic symphony. In that time, life has transformed from invisible microbes drifting in ancient seas to the immense diversity that now covers the planet. Each living creature, from the smallest insect to the largest whale, is a single verse in that unending song, shaped by both inheritance and experience, carrying forward the legacy of those who came before. At the heart of evolution lies variation, the subtle differences between individuals of the same kind. These differences arise from mutations in DNA, small changes in the sequence of genes that may alter the way a body grows, functions, or responds to its surroundings. Most of these changes are neutral or silent, passing unnoticed through generations. But occasionally, one offers an advantage: a slightly stronger muscle, a sharper eye, a tolerance to cold, or a quicker mind. When such a trait increases an organism's chances of survival and reproduction, it becomes more common in the population. Over time, these small advantages accumulate, and the species itself begins to change. This quiet, natural process is what Charles Darwin called natural selection, the gradual refinement of life through adaptation. Natural selection is often misunderstood as a harsh or competitive struggle, but it is, in truth, a delicate balance between life and circumstance. Organisms are not engaged in constant conflict. Rather, they are responding to the world around them, adjusting to its conditions. A bird's beak grows longer not out of will or rivalry, but because those with longer beaks can better reach food, and thus leave more offspring. A plant spreads its seeds farther not out of strategy, but because the wind carries some a little further than others, and in that randomness, new possibilities emerge. Evolution is not guided by intention, it is guided by persistence. What works remains, what fails fades away. And through this gentle filtering, the forms of life shift across time. Over millions of years, these gradual changes have produced astonishing results. From the earliest single-celled organisms came multicellular life cells learning to cooperate, to divide labor, to specialize. Some became tissues, others organs, each serving the greater whole. In the oceans, fish evolved fins that allowed them to swim more efficiently, and later, some of those fins transformed into limbs that carried the first amphibians onto land. Reptiles, in turn, gave rise to birds, their scales reshaped into feathers, their arms into wings. Mammals emerged, small and warm-blooded, able to thrive in changing climates. From them came primates, with grasping hands and curious eyes, and from primates, eventually, the human being, capable of looking back and wondering how it all began. But evolution did not end with us. It continues still, silently, in every corner of the earth. It continues in the bacteria that learn to resist antibiotics, in the flowers that adapt to new pollinators, in the animals that change their patterns as the climate shifts. The story of life is ongoing, and we are not its final chapter. Evolution is not a ladder with humans at the top. It is a vast, branching tree with countless paths, each one equally valid, each shaped by its own needs and circumstances. In this tree, no species stands apart. All are connected through shared ancestry, their roots entwined in the same ancient soil. Fossils, the imprints of life's past, offer glimpses into this great unfolding. In layers of rock, we find the bones of creatures that no longer exist, but whose forms hint at transitions: fish with the beginnings of limbs, reptiles with traces of feathers, early mammals small and nocturnal beneath the reign of dinosaurs. Each fossil is a page from an ancient book written in stone, telling of adaptation, of extinction, of renewal. The Earth's surface itself is a vast archive of change, holding the memory of evolution within its depths. Even within our own bodies, Traces of that history remain. The bones of our hands echo the fins of fish. The structure of our lungs recalls the swim bladders of ancient aquatic ancestors. We carry the story of evolution within our very cells. Yet evolution is not only about physical change. It is also about the emergence of complexity and consciousness. As nervous systems developed, perception and awareness evolved alongside them. The first spark of sensation, a cell reacting to light or touch, eventually became the capacity for experience, for memory, for thought. The human brain, with its billions of interconnected neurons, is perhaps evolution's most intricate creation, not because it is superior, but because it is capable of reflection. Through us, evolution has gained the ability to contemplate itself. We are the universe's awareness of its own unfolding, matter becoming mindful of matter, life gazing back upon its origins in wonder. There is a profound humility in this realization. Evolution reminds us that we are not separate from nature, but part of its continual transformation. The atoms in our bodies have passed through countless other forms through plants, animals, and oceans before coming together in us. We are not masters of the world, but participants in its ongoing story. Our consciousness, remarkable though it is built upon the same principles that govern the flight of birds and the growth of trees. To live with this understanding is to live in harmony with time itself, to see that change is not to be feared, but embraced, for it is the essence of life. Evolution also teaches us that fragility and resilience coexist. Many species have vanished, unable to adapt to shifting conditions, while others have flourished in their wake. Extinction, though it seems tragic, is part of the same process that gives rise to new life. The earth renews itself through cycles of loss and creation, shaping each generation from the remains of the last. Even now, evolution continues to respond to the pressures of a changing planet, climate, movement, and chance. The story of life is not one of permanence, but of persistence through transformation. In the quiet language of genetics, evolution writes its poetry. Every living thing carries in its DNA the memory of what has come before, a vast library of adaptations, experiments, and connections. When a cell divides, it passes that library on, with small variations that keep the story moving. Each organism is both author and reader, writing the next line of a book that will never end. The beauty of this truth lies in its gentleness. Life does not seek to dominate, only to continue, to explore what is possible within the boundaries of the physical world. To contemplate evolution is to see time not as a force that destroys, but as one that creates. Every wave that erodes the shore also shapes it. Every change that seems like loss gives rise to something new. Evolution is the patient hand of the universe, sculpting life from dust and energy, from water and sunlight. It is the quiet flow of adaptation, unhurried and unending, the way the earth breathes and renews itself. We, too, are part of that flow. The same process that shaped the wings of birds shaped the hands that now write and build and reach toward understanding. The same slow unfolding that formed coral reefs and forests has given rise to thought and emotion. Evolution is not something that happened long ago. It is happening still, in every heartbeat, in every cell, in every new generation that carries life forward. It is the endless adaptation, the gentle pulse of transformation, through which the universe continues to know itself. 20. The unity of life, the web, that connects all things. If one could step back far enough to see life not in fragments but in its entirety, the divisions we so often perceive between species, between forests and oceans, even between ourselves and the earth would begin to fade. Beneath the differences of form, color, and sound, there runs a single, unbroken thread, connecting all living things. This thread is not metaphorical but material, woven from the same molecules. Life, in all its diversity, is one continuous fabric, a vast web where every organism, every cell, and every breath is joined in quiet partnership with every other. The unity of life is not an idea to be imagined. It is a truth that can be traced in the very chemistry of our being. At the most fundamental level, every living creature is built from the same elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, arranged into the same essential molecules: proteins, nucleic acids, lipids, and carbohydrates. The DNA that shapes a human being is composed of the same four bases that guide a blade of grass, a whale, or a hummingbird. The genetic code is universal, a language shared by all life, written once in the deep history of the earth and spoken ever since in countless forms. Whether in a cell dividing in a leaf or one beating in a human heart, the same processes unfold. Energy flows, molecules combine, enzymes guide, and life continues. The differences that separate species are small when compared to the vast sameness that binds them. This unity extends beyond the molecular into the patterns and systems that govern all living things. Every organism, no matter how simple or complex, depends upon the same cycles of matter and energy. The carbon that forms a tree's trunk may one day pass into the air to be taken up by another plant or inhaled by an animal. The water that flows through a river is the same water that circulates within our veins. The oxygen, produced by a single cell of algae in the ocean, may be the same molecule that sustains a breath taken miles away. The entire biosphere, the thin, fragile layer of life that surrounds the Earth, is a single, interconnected system, its parts inseparable from one another. The relationships between living beings form the delicate web of ecology, the living network of mutual dependence that sustains the planet. No creature exists alone. The bee depends on the flower for nectar, and the flower depends on the bee for pollination. The predator relies on the prey, and the prey on the plants that feed it. Even the smallest bacterium contributes to this balance, breaking down waste, releasing nutrients, and keeping the soil fertile. Through these exchanges, life sustains itself, an endless circulation of giving and receiving, of nourishment and renewal. This web is not static. It shifts and adapts with the changing of the seasons, with the rise and fall of populations, and with the slow turning of time. In every ecosystem, from rainforest to desert, from coral reef to tundra, this interconnection reveals itself in quiet harmony. When one part of the web is disturbed, the effects ripple outward, touching even the most distant corners of the system. A single missing species can alter the flow of energy and the rhythm of growth. Yet, remarkably, nature seeks balance again and again. New relationships form, and the web reweaves itself in endless resilience. This adaptability, this ability to restore harmony, is one of life's greatest strengths. It reflects not competition, but cooperation, a shared persistence that transcends individual forms. Even within the body of a single organism, this pattern of unity repeats itself. Trillions of cells work together to create a coherent whole, each performing its own task, yet dependent on others for survival. Muscle cells contract, neurons transmit, red blood cells carry oxygen, and immune cells defend. None could survive in isolation, but together they form a living system, capable of thought, feeling, and action. The human body is, in this sense, a microcosm of the planet, an ecosystem of its own, where cooperation and balance maintain the harmony of life. Within us live not only our own cells, but countless microbes, bacteria, and fungi that help digest food, protect against disease, and even influence mood. We are not singular beings, but living communities, woven from many lives intertwined. The unity of life also stretches through time. connecting all beings not just in the present but across generations. Every living thing on Earth shares a common ancestor, a single cell that lived more than 3 billion years ago. From that cell, through countless divisions and mutations, every species has descended: the trees that shade our paths, the birds that fly above, the whales in the depths, the people who walk the earth, all trace their lineage back to that first spark of life. The diversity we see today is the result of branching from that single origin, an endless unfolding of form and function. To know this is to see that we are not separate from the world but its continuation, the latest expression of a process that began long before memory and will continue long after we are gone. In this light, individuality becomes part of a larger continuity. The boundaries we perceive between one organism and another, between living and non-living, are not fixed but fluid. At the atomic level, the particles that make up our bodies are in constant exchange with the world around us. With each breath, we inhale molecules once part of ancient trees or distant stars. The calcium in our bones was forged in stellar furnaces billions of years ago. The iron in our blood once flowed in the core of a dying Sunday. Even now, atoms leave our bodies and enter others, moving through air and water, through soil and root, returning again in different forms. Life is not a possession but a shared inheritance, a single current flowing through all beings. To recognize this unity is to find peace in the constant motion of existence. The world is not divided into living and dead, self and other, but joined in a vast continuum of energy and matter. Each being, no matter how small, plays a role in the great equilibrium of life. The moss on a stone, the insect in the air, the human thought, all are expressions of the same universal principle: the desire to endure, to connect, to evolve. This awareness brings humility, for it reminds us that we are neither masters of the earth nor separate from it, but participants in its living fabric. Every act of kindness, every breath of gratitude strengthens that fabric, binding us closer to the whole. In the unity of life, there is also a deep beauty, the understanding that diversity itself is the form through which oneness expresses creativity. The forest thrives because no two leaves are the same. The ocean endures because it contains multitudes. Just as cells within a body differ yet cooperate, species within an ecosystem coexist and support one another. The variations that evolution produces are not divisions but harmonies, different notes in the same symphony of being. The richness of life lies in its multiplicity, in the endless ways it finds to express a single truth, that all is connected. When we gaze at the natural world with this understanding, even the simplest things become sacred. The pattern of veins in a leaf mirrors the branching of rivers, the network of neurons in a brain, the structure of galaxies in the cosmos. These repeating forms speak of an underlying order that unites all scales of existence. The same geometry, the same rhythm, flows through everything, from the spiral of a seashell to the spiral of a star. Life is not separate from the universe but its living face, the means by which it observes and celebrates its own unfolding. In the end, the unity of life is not something to be proven, but something to be felt in the stillness of a forest, in the rhythm of waves, in the quiet beat of one's own heart. When we breathe, we participate in a process older than time, shared by every living being. When we walk upon the earth, we move across the same ground that has given rise to all existence. The web of life surrounds us, within us and beyond us, infinite and whole. To live with awareness of this unity is to live gently, to act with reverence, to recognize that in caring for any part of the world, we care for the whole. For in truth, there is no separation, only the endless continuity of being, the eternal web that connects all things.